Welcome to Module 2. This video is going to be on biomolecules. Molecules important to biology. And here are the 4 categories that we're going to look at today. As usual, here are our learning objectives. You can use these as you are studying. This is what we're going to go through in today's video. The first topic to say and go through is what are organic molecules? Carbon, remember the elements we talked about last time, that periodic table. Carbon is actually a very important element because it's the most abundant element in all living systems. Why do we care about this? We are living humans, right? And so carbon is the main component of what we call organic molecules. Organic molecules include what we call biomolecules. Biomolecules, they are molecules important for biology and making up these living organisms, right? Anything living is made up mostly of these biomolecules over here. The biomolecules we'll talk about are carbohydrates. This, we'll talk about different sugars. You might have heard a bit of carbs, right? Carbohydrates. We are made of carbohydrates, partially. Proteins make up the human body as well. You've probably heard about proteins, especially with muscles, but they're in many other places too. Nucleic acids. This one might be one that you're new to. You might have heard of a molecule called deoxyribonucleic acid, which has an abbreviation, a cool nickname called DNA, nucleic acid, DNA. So we'll talk about DNA. And human bodies are also made out of lipids. Lipids contain different fats. We'll see phospholipids. We'll talk about steroids. These fit into this category over here. These 4 categories I want you to know exist, and this is the 4. Let's see them as the 4 building blocks of the human body, right? We use these Legos to build up cells and human bodies. So, what are organic molecules? Organic compounds are any molecule with linked carbon and hydrogen atoms. Okay, that's the definition of an organic compound. You might have heard of organic chemistry even, right? Like that is a field of chemistry to study these molecules that are important to living organisms. Carbon is actually a flexible building block, we call it, because it can make many different arrangements. Atoms make different bonds, and some of them can only make 1 bond or 2, right? Carbon actually can make 4 different bonds. It's kind of like in a Lego set, you have some of those connector pieces that could really help you branch out your little Lego set. Well, that's what carbon is, right? It's an atom that can really have different bonds put to it. So carbon, we call it the backbone of organic molecules, okay? So carbs, proteins, nucleic acids, lipids, they all have as a backbone carbon. These carbon and hydrogens that are repeating, right? Remember the definition of organic compounds are that they're linked carbon and hydrogen atoms, right? So carbon backbones of organic molecules can vary in their arrangement. They can be long linear patterns with or without some double bonds. So here you have 1, 2, 3, 4 carbons all linked with hydrogens. You'll note that they all need 4 bonds, carbons. Over here, you'll see that there's a double bond here. And remember, carbon can only have 4 bonds, so 1, 2, 3, 4. 1, 2, 3, 4. Each of these carbons still has 4 bonds. Here you see that the double bond is actually in the middle of the molecule, right? These are all different types of molecules that could exist. Also, this is, I told you, it's like a Lego piece, right? Like over here, you have 3 carbons, and then you have 1 carbon that's linked over here. If you count all the bonds for the carbon, they still all have 4 bonds, right? But this carbon in the middle here is a good Lego connector piece so that you could branch out your chain over here, right? So in this picture here, you see that you can make rings of carbon, right? So you can make these little rings, and carbon still will have 4 bonds, or you could make rings with double bonds over here. All this to say, this is some crazy chemistry, but all this to say that carbon, right, is capable of having these different arrangement patterns, right? And so organic compounds, they have a backbone of carbon and hydrogen, and they'll then add different branches, different groups to that, and they'll really be able to have a variety of interesting molecules that can sustain and create life, right? Inorganic compounds do not contain carbon-hydrogen bonds. So for example, this inorganic phosphate, I do not see any carbon or hydrogen bonds, right? So this is an inorganic compound. It doesn't mean we don't have it in our body, right? But remember what I said, organic compounds, they are the main component of living systems, right? So they make up a big chunk of our bodies. And so these carbohydrates, proteins, nucleic acids, and lipids, they are considered organic compounds. We're going to see that they all have these carbons and hydrogens, okay? I don't want you to get scared by the chemistry. I just want you to start recognizing some patterns. Oh, this kind of looks like a carbohydrate. This looks like a protein, nucleic acid, or a lipid. All right, and I'll start teaching you how to do that. So I want you to pause the video and circle the organic molecules below. There may be more than one. Okay, so over here for A, right? You have a carbon and it's linked to 1 hydrogen, 2, 3, 4. Our definition of an organic molecule is that there are CH bonds, right? So this is definitely an organic molecule. This is 1 hydrogen linked to an oxygen, linked to another hydrogen. That's actually H2O, water. This is in our body. A lot of it is in the human body, right? But it is not an organic molecule. This does not fit in that category. CO, do you see this CO double bond, CO double bond? This is CO2. Aha, CO2. This is carbon dioxide, 2 oxygens, carbon dioxide. CO2 is a gas that we exhale from the wastes we put in our body, right? So we exhale out CO2 after our cells process the oxygen that we bring in. So this has CO bonds, but remember our definition, our strict definition is that organic compounds are any molecule with linked carbon and hydrogen. So this would not fit our definition. This over here, CH bonds, there's so many different CH bonds over here, so this definitely fits our definition. And lastly, over here we see some CH bonds, right? And we see a little additional OH on the side. This usually makes this an alcohol when there's an OH, but it's still an organic compound because in it we find these CH bonds. So this would fit our definition of an organic compound. Why am I doing this? Because I want you, when you see some crazy letters that make up a molecule, you'll know if it's organic or not. And then I'll give you some more clues on how to define whether it's a carb, a protein, a lipid, nucleic acid. We're going to go through that. So that's our little activity here. Okay, so this is what I was talking to you about. Biomolecules are organic molecules essential to life, right? So these biomolecules are the ones important for our course, biology. right? Biomolecules and organic molecules, there are little differences between them, but in essence, right, they're kind of, these are the ones that are important for the human body, that make up our human body. And I want you to know one general function for each, okay? And soon one general, like the structure of the general idea of the structure of that, okay? So, first biomolecule in the human body are carbohydrates, right? Has carbon in the name. Hydrates sounds like hydrating, like water. We're going to see that in a second. Carbohydrates, right? Do you see these hexagons all linked together? This is a big carbohydrate, a big sugar. These provide a source of short-term energy to cells. Our cells use them to get fuel, right? When you eat carbohydrates, when you eat sugars, you can get short-term energy. You could get even a sugar high, but full of sugar, right? So these are carbohydrates. They act as the hydroquebec of the cell. They act as the energy source, the short-term energy source to get some fuel, right? Sometimes we say we fuel the cells, we give them some energy, we give them carbohydrates, okay? Proteins, this is a wacky ribbon, right? Looks like something that's on a present. All these little ribbons over here, right? These are, we're going to see what a protein is soon, but these proteins are these folded up in weird shapes, these little groupings. These are the workers of the cell, right? You maybe heard of proteins for muscles. That's very important for muscle contraction, but they're even more important than that. Every single cell, it's not just in muscles that you find proteins. Every single cell has these proteins that we consider the workers of the cells. They provide structural support. They build up certain networks inside the cell. They transport molecules. They are little bridges across the cell. They are the little transporters that bring things in and out. They speed up reactions. These are called enzymes. These proteins, they are the workers. They're converting things inside the cells to make new molecules that are needed. They build up molecules, break down molecules. So proteins are essential, right? They act as bridges for transport, and they are the workers of the cell. And you know them in terms of muscle contraction. We'll talk about that later on. Nucleic acids in every single cell, right, for the most part, are nucleic acids, which, again, nucleic acids, you probably heard of deoxynucleic acid, DNA, that encodes and transmits genetic information. It is the instructions of the cell, how to build proteins, how to build the workers, right? We know that the workers, right, they are affected by the laws of the parliament, right? So the parliament makes some laws, makes new jobs, makes all of these things, and dictates kind of how the work is done in a country. Well, the DNA, right, the nucleic acids, they have the genetic info which encode and they can encode different proteins to be built, or kind of guide the cell into how to function, okay? And we keep the instructions of the DNA hidden in the cell. We'll see that today in the nucleus. That's important. Lipids make up cell membranes. So every single cell, if I cut a cell in half, do you see these little, they look like toothpicks, these little molecules over here that make up the surrounding of the sphere, kind of like the outside of a balloon, right? These little lipids, right, they make up the cell membranes, and they provide long-term energy to the cells. So These are two separate functions. These types of lipids here, they are the ones that make up the cell membranes. Other types of lipids called neutral fats or triglycerides, you know that fat could be stored inside of us, right? And that's actually very useful for long-term energy, right? Not too much of it, but triglycerides, right? We do need them to have long-term reserves in case, you know, that when we can't always be eating carbohydrates constantly, this short-term energy. So these lipids, these fats are good for the long term. If we don't eat for a little while, we can break down some fat reserves, right? So, in terms of the cell membrane idea of the lipids, right, they act as the border agents of the cell, right? They control what goes into the cell or not go into the cell, right? So I've just glanced over the idea of the four categories. I need you to know these quite well. And we will delve into each of them in a humongous amount of depth. But I find in biology, if ever you get lost, right, just know one general function for each thing. So if you're losing and disorganized in what is what, I would suggest that you take a step back. Okay, what is a carb and what is its function? What is the protein and what is its function? What are the nucleic acids and one general function? And it'll help to, you know, just get a big picture idea. Now we're delving a little bit deeper. But before we do that, these biomolecules, carbs, proteins, nucleic acids, they are made up of smaller bits, right? And that create these bigger bits. So monomers are the building blocks that form larger polymers in biomolecules. Okay, so the biomolecules, you'll notice, look at this carb, it has many hexagons. These little hexagons we'll talk about in a bit, but those are monomers, right? And they form this big polymer, okay? Maybe you've heard of complex sugars and simple sugars, right? Like there's smaller bits and bigger bits. I want you to be aware that the major macromolecules, the big molecules, the biomolecules, they are mostly polymers in our body, right? They're complex molecules that are made up of repeated simpler monomers, okay? And they're connected by bonds, all right? So do you see these monomers, these little bits here? They can link together to form this big polymer. Mono means one, right? And poly means many, right? So monomers come together, bond together to form big polymers. All right. So monomers vary based on the type of biomolecule polymers, right? So monomers and polymers, it's umbrella term. It's like saying the small pieces and the big pieces. What are the small pieces? Well, it depends on what specific biomolecule you're talking about. So let's look at the monomers for sugars, right? For the carbohydrates. The monomers are called monosaccharides. Saccharide means sugar, I believe, in Greek. So mono, one, saccharide, sugar. These little hexagons are monosaccharides, right? They're simple sugars. They're separate simple sugars. If you piece them together, they form what we normally call a long carbohydrate, right? So this is a polymer, right? A long chain, but we call it a carbohydrate or we call it a polysaccharide. Many saccharides, many monomers put together, right? Many little monosaccharides that we link together to form a polysaccharide, right? So this is the term for the sugars, monosaccharide and polysaccharide. Either you have one unit or you have many of them linked together. For proteins, the terms are a bit different. The smaller monomers are called amino acids, And we're going to see why that's called like that soon. It's annoying, I know. Amino acids are the smaller bits, and they come together and they form what we call a protein. Okay, so the monomers are called amino acids, and the polymers are called proteins. Some people call them, and we'll get to this in a bit, some people call them polypeptides because these are peptide chain bonds that link together the amino acids. So many peptide bonds form together to form this big protein, and we'll talk about that in a bit. As I said before, biology is a lot of terms, it's overwhelming. I'm throwing this all at you. And this is the slide to review at the end. But we're going to go through each of these separately. Right now, what I want you to understand is that smaller monomers come together to form polymers in each of the biomolecules. In sugars, the monomer is called a monosaccharide, and it comes together to form a polysaccharide. In proteins, we see that amino acid monomers come together to form polypeptide polymers, right? In DNA, we're going to see that nucleotides, right? Nucleotides, maybe you remember this from high school, A, T, C's, and G's, right? We'll talk about those when we get there. They come together and form a chain of this weird sequence of DNA, right? They form this nucleic acid, this DNA, or we'll talk about RNA also. So that's nucleotides forming nucleic acids. And then we have fatty acids and glycerol, right? This is 3 fatty acid chains. They're long chains of carbon, and a glycerol molecule. They come together and they can form this triglyceride, which is a type of lipid. I want you to note that not always is a polymer made of repeating monomers. Lipids have like a weird exception, but they are well organized regardless in smaller bits and bigger bits. There's a bit of a definition specificity that I don't think we need to dwell on too much. But yeah, this is the how we make up this fat molecule. This is what you store in fat cells in your body. So little bits, these 3 fatty acid chains and the glycerol come together to form lipids. I just threw so much at you, right? But I want you to understand, what I want you to get from this is that small monomers piece together and form polymers, right? And then for each of the type of biomolecule, they have a specific name, right? These are all monomers, right? But depends, they have different names depending on what biomolecule it is. And these are all polymers, but they all have different names depending on what biomolecule they are. Monomers just means small bits. Polymers means longer bits that are chained together. So how are polymers built and broken down? Monomers are built to form polymers. How is this done, right? So think of it in the human body. Before we go into this, we eat food, we actually break it down into smaller monomers in our digestive tract, and we absorb it into our blood. We absorb little monomers, right? And then we need to use those to build the stuff that we want. We build polymers, right? So how do monomers form a polymer? This process is called a dehydration synthesis reaction. What the hell does this mean? Dehydration means to remove a water molecule. Synthesis means to build something, right? To build up something. Synthesize something. You're going to hear this term a lot, so you should know that synthesis means to build. And this is a reaction, right? So to turn monomers into polymers, you need to build. To take these little blocks and build a house, a big chain, right? You need to build things, right? So you synthesize, you build, and it's dehydrating. You need to remove a water molecule. So what I'm going to show you is that when we do a dehydration synthesis reaction, we actually take 2 monomers and or two chains that are separate, right? And we bond them together through a loss of a water molecule. Do you see over here, this chain has 1, 2, 3 monomers, right? And on every chain, there's always an OH side and there's always a hydrogen on the other side, right? Over here, you see this monomer. It's one monomer long. It has an OH side and an H side. These two sides join together, they bond, but they release the H and the O, and this H come together, right? And they bond to form a water, and the molecule gets dehydrated and forms one water molecule in excess, right? It bonds together and it releases this water molecule, and now the chain is formed, right? We have 1, 2, 3, 4 of these monomers put together. This is called the dehydration synthesis. We dehydrated the molecule, right? We removed a water molecule. We actually created a new water molecule from the binding of these two, right? And we synthesized. This molecule is now synthesized together. It comes together into these 4. This was 3, this was 1, and it became 4. 3 plus 1 makes 4. So this is now a polymer with 4 little monomers on it. We synthesized a longer polymer, right? Now, polymers, right, these long chains are sometimes broken down into monomers. I'll give you some examples of where we do this. In the human body, when you eat food, you eat a lot of complex polymers from either animals or plants, and then you break them down and digest them to smaller monomers, right? Or even in the human body, sometimes we need to break down molecules that are no longer good, or many, many different things, right? But these polymers sometimes need to be broken down to monomers, and there's reasons why we need to do that. So when we do that, polymers, they are disassembled to monomers by what we call a hydrolysis reaction. I want you to study this word carefully. Hydro means water. Lysis means to cut. To lyse something, lyser quelque chose, means to lyse and cut. You're going to see this word come up a lot, right? So water lysing, water cutting, right? You will need to introduce a water molecule to cut the bond, okay? So polymers are split apart through the addition of a water molecule. Here, 1, 2, 3, 4, right? There are 4 little monomers in this big polymer. And we introduce a water molecule into this bond here, and it will lyse the molecule into 2. Do you see how here we created a water molecule, and here we need to introduce a water molecule into the molecule to lyse it, right? So this is how polymers are built, through dehydration, synthesis reactions. And this is how we break them down, through a hydrolysis reaction. We introduce water so that I could split the bond apart into 2. So, like this picture over here is very, this is very, just the general idea, but now I want to apply it to our biomolecules. Remember, we said there's all these biomolecules. Some are monomers, some are polymers. Now I'm going to piece the 2 ideas together, okay? So monomers on this side, polymers on this side. I'm going to give you a few examples. Do you see this monosaccharide here? That is like a simple sugar, like one little sugar, right? Monosaccharide 1. This is another monosaccharide. They look like hexagons. They have carbons that are in a hexagon shape. So this monosaccharide, right, is not linked to this one. There's a little blue square around it just to show you what bond is going to come together, but the atoms here are not linked together, right? If I follow this way, this separate monosaccharide and this separate monosaccharide, they will come together and form look at this, a polysaccharide, right? If you look at the math, however, or the number of atoms, this OH and this H of the other molecule came together, bonded, and was removed, right? And it formed this little O over here, stayed over, and formed this polysaccharide. That's a dehydration synthesis reaction. We took the OH on this monosaccharide, the H on this one. It formed a water, it dehydrated a water molecule out of this joining together, and it created a new water in the environment, right? And we formed this polysaccharide, right? We formed this this polymer that has two saccharides together. So that's the definition of a dehydration synthesis reaction when you form together this over here. Look at this. Here there was a piece with an H. Here there was a piece with an OH. We removed the water and we formed this polysaccharide or this polymer, right? Here I had an OH, here I had an H. They got removed in the bonding, right? And instead we now have this bond over here that's now connected. The opposite is true. I can take this polysaccharide, right? This is why the arrow is a bit weird here. Forget about hydrolysis for now. You saw that as we went in this direction, H2O was removed and it formed this polysaccharide. Now let's focus on the bottom arrow. If we go from here, the polysaccharide, to this side where we have separate smaller monomers, smaller sugars, these monosaccharides, right? Then this is called a hydrolysis reaction. Hydro means water, lysis. Water is introduced over here actually. Water is brought in over here. And one OH will go on to this side and an H will go on to this side with the O that's already there. And it'll form these two separate monosaccharides. So we can go from this side to this side and break them apart through what we call a hydrolysis reaction. You know this. When we have two Lego blocks, you can piece them together to form a chain. And then a minute later, you can decide to take those two Lego blocks and unpiece them together. That's what this is, okay? Now, over here, let's look at the same principle. This looks a bit different than our sugar, right? This is called an amino acid. It's the building blocks of proteins. This is one amino acid, and this is a second amino acid. And do you see over here the O and the H, right? They're going to come together and dehydrate. They're going to remove the water and create a water in the environment, and they will form this bonded together protein. This protein has 1 and 2 pieces, Legos, that came together to form a longer molecule. I know there's weird letters everywhere. We're not chemists. Don't worry too much about it. I'll tell you what we need to know. I just want you to get the big picture of the Lego blocks and not be scared of the letters. The opposite is true. Sometimes we break down proteins. Let's say your muscles start breaking down, right? This is like forming a muscle. 2 amino acids, right? While you're working out, they came together. They removed an an H2O, a water molecule, and they formed a bond to make a bigger, longer muscle protein, right? To make a big muscle. Then if you break down your muscle, right? You stop going to the gym for a bit. These two little pieces here, right? The peptide bond in between can be broken by introducing a water molecule, hydrolysis. You could introduce a water molecule here to the peptide bond and lyse the two molecules and break the molecule that was 2 pieces into their separate pieces. So we go from the polymer, the protein, to the 2 separate amino acids. That's called hydrolysis, which is called hydrolysis, right? You're lysing with a water molecule. Okay, now over here, right? Here's one nucleotide and here's another nucleotide. When they come together, right? dehydration, synthesis, synthesize together. The water molecule, again, escapes, right? It dehydrates from the molecule, the nucleotides, and it forms a new water. This will link together and form a nucleic acid, and maybe even soon enough, a DNA, a deoxynucleic acid, right? This is what a weird DNA looks like. If you go the other way around, sometimes DNA is broken apart. So if you break this bond over here, you introduce water into this bond, then you will have hydrolysis, right? Hydrolysis. Water will come in and chop this bond and go onto each of these sides, right? That's hydrolysis. Don't get scared by this diagram. I just want you to appreciate you could go to one side or you could go to the other side, right? You could go in either direction. All right, so now we start our journey. This is a general concept that sometimes your biomolecules, first of all, what I want you to get from this is to remember that for each of these biomolecules, there's separate monomers. And for each of these biomolecules, there's different names for the polymers, depending on what biomolecule it is, right? We're going to need to know that going forward. So this is a good slide to look at. This slide here is important because for every one of these biomolecules, the same thing can happen. You could build a chain of Legos together, or you could break the chain down. And our body is just a bunch of reactions building and breaking things, okay? So I need you to understand this concept pretty well. And now we're going to look at it for each of these separately. So for carbohydrates, right? What are carbohydrates made from? Carbohydrates are made of carbon, hydrogen, oxygen, in multiples of C1H2O1. Okay, so a carbohydrate could be C1H2O1. It could be C2H4O2. It could be C3H6O3, right? As long as the H is doubled, then it's considered a carbohydrate. So it's always in these proportions, and it's because of the ring formation and a bit of chemistry that they follow this pattern. So look at the name. Don't memorize things. Always look at the name. Carbo equals carbon, right? So these are made of carbon. Hydrate sounds like water, right? So it sounds like H2O. These are made of carbons and hydrates, water, right? Carbohydrates are also referred to as saccharides sometimes. Monosaccharides, polysaccharides. This is the Greek word for sugar, actually. So we'll take a look at this in a bit. Monosaccharides, also you might have heard the term simple sugars, right? So this is a monosaccharide. We draw it as a hexagon, but each of these corners is actually a little carbon molecule that's there. This is one single carbohydrate unit, or we also, remember we said that this was a monomer, right? This is a one little Lego block. This is a monosaccharide, one Lego block. And if we put two Lego blocks together, two monosaccharides together, right? This is called a disaccharide. It's kind of the in-between. So disaccharide is two linked monosaccharides. And then anything, well, there's other terms, but bigger things, right, are called polysaccharides. They're complex sugars. Maybe you heard of complex sugars. These are many linked together monosaccharides, long chains of linked monosaccharides. These are called polysaccharides, right? So here's our monomer, here was our polymer, and here's a little in between. So let's start with carbohydrate monomers. This is the monosaccharide. This is the simple sugar, one hexagon, right? Carbohydrate monomers are called monosaccharides. Mono, one, saccharide in Greek means sugar, right? Saccharin, maybe you've heard of. So monosaccharides are what we call a hexose sugar. They are 6 carbons, all with the formula C6H12O6, right? Remember I told you that the formula is C1H2O1? It could be any multiple of this. So it could be C2 H4O2, right? Or C6H12O6. Well, I'm telling you that all monosaccharides, all the simpler units of sugar are hexagons, right? So this must mean that it's C6H12O6, right? It's 6 carbons because it makes up each of the little each of the corners of this, right? So over here, we're going to look at the 3 types of monosaccharides that we have. We have glucose, okay? We didn't teach you about chemistry is that in this little hexagon here, right? Each of the corners here is actually a carbon, right? So 1 carbon, 2, 3, 4, 5, 6 over here, right? This is an oxygen in the corner. This is the same thing. If I chopped this molecule and spread it out long, then it would look like this. You can count 6 carbons as well, right? If you don't believe me, for fun, you can go in this molecule and count how many carbons, hydrogens, oxygens there are. It should come out to C6H12 and O6, right? So this molecule here is called a glucose. Okay, it's a hexose because it's a hexagon, right? And this is a monosaccharide. This is a building block. This is like a red Lego block, let's say. Okay, fructose still has 6 carbons. 1, 2, 3, 4, 5, 6. It has 6, but the arrangement, you'll notice, they arranged it in a different way. This is like a blue Lego block, let's say. And galactose is another arrangement. It's carbon, 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 carbon. There's 6, right? But the only difference here, I believe, is the orientation over here of the OH on top versus being on the bottom over here compared to the glucose. And that actually does all the difference. But we're not chemists, right? So what I want you to notice is all of these monosaccharides are the building blocks. This is the red Lego block, this is the blue, this is the green, but they're all the baby blocks, right? They all are the same size, they're small, but they're different colors. They're a little bit different in their orientation, right? But they all have the formula C6H12O6. They all have the same size, right? They're the baby ones. And we can link them together to form other things. And we'll talk about that in a bit. So hexose monosaccharides, they could be drawn as linear skeletons, right? Sometimes we can draw them like this. For example, glucose, C6H12O6 looks like this. But the truth is, in an aqueous solution, aqueous, aqua, watery solution, they actually assume what we call a ring configuration, right? In a solution, when you put it in water, in a watery substance, which our whole body, human body and cells are water, bathing in water, right? This glucose actually will bend and form this ring with little flags sticking up and out of it, right? This is a ring configuration. This is a glucose molecule. So that's why we draw them as these rings, because in the human body, when they're in watery substances, which is almost all the time, they form these structures, okay? So whenever you see these little hexagons, right, or pentagons, you know that this is a monosaccharide. If you see one, we draw it like this, but the chemistry of it is actually this, right? Don't freak out. Just know that, if it's in this shape, right, this hexagon, I know that this is probably a monosaccharide, right? All of these, just looking at them one at a time, you should be able to say, that's a monosaccharide, right? And why are monosaccharides important? Their function, right? Glucose is the most important one. it is a source of energy for animals. Remember what we said about carbs? It's a short-term energy. Glucose is needed for almost all cells of the body, and glucose is needed because with oxygen that we breathe in, the mitochondria in almost every single cell, right, will convert these two things into CO2, water, and energy. It's called ATP. We'll see that in a bit. We need glucose as fuel. You know this. When you're tired and you eat a lot of candy, you could get a sugar high, right? Because glucose is fuel for your cells. It gives energy to every single cell. If you don't eat, you die, right? So you need glucose in your body and oxygen, right? And that will make energy. When we light up a fire to make flames, right? All the sugars that are found in the wood or in the paper that you're burning, right? All of those are getting burnt down to form this heat energy, right? So sugars, glucose is a controlled fire inside of us. It's a fuel. It's a source of energy short term that we can use, okay? So in this picture, I want you to go and identify the monosaccharides. I will give you a few seconds to do so. You can pause the video and go ahead and do this. All right. If you're done, right, I want us to go and take a look at this. Remember our definition of monosaccharides. They're hexose, 6 carbons with this formula. They must have this formula, C6H12O6. So let me write it here, C6H12O6, right? So over here, I want to count the carbons. I have 1 carbon. I have 2 carbons. I have 3 carbons, 4 carbons, 5 carbons. Oh, nope. This is not a monosaccharide, right? Over here, I have 1 carbon, 2 carbons, 3 carbons. That's not a monosaccharide, right? Over here, I have 1 carbon. I have 2 carbons, 3. This is not a monosaccharide, right? Here I have 1 carbon, 2 carbons, 3, 4, 5, and 6, right? And there are probably 12 H's and 6 oxygen. This is going to be my monosaccharide, okay? You can go ahead and count the other ones. I'm sure they're all good. Usually when you have a good balance of one, then the other one follows, all right? Let's do it for fun. So 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. And oxygen, 1, 2, 3, 4, 5, 6. There we go. Over here, I have 1 carbon, 2 carbons, 3, 4, 5, 6, 7. Whoa, way too much. Not a monosaccharide. Do you see how here sometimes they're in linear formation or in the ring, right? So sometimes they're trying to confuse you, like this was in the linear formation. So don't fall for some traps here. Just count the number of carbons. So 1, 2, 3, 4, 5, 6. Okay, we're good. Let's count the hydrogens. 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Oxygens. 1, 2, 3, 4, 5, 6. So this is a monosaccharide, right? Do you see how this one's drawn in ring form and this one's in linear form? It's the same thing, right? It's just that they're drawn in different ways. So I just want to reiterate something. The key takeaway is that the monomers for carbohydrates are called monosaccharides. They look like these little hexagons, right? And there's actually 3 kinds of monosaccharides. There's glucose, fructose, or galactose. Those are the subtypes of monosaccharides. Okay. Disaccharides. These are carbohydrate intermediates. They're the middleman between a monosaccharide and the polysaccharide. Okay, so I call these the intermediates because they're kind of in between a mono and a polysaccharide. Okay, they're disaccharides. They're still small, but they're just 2 units. So let's take a look at this carbohydrate intermediate, the disaccharide, which has 2 together, right? 2 monosaccharides together. Disaccharides are formed via what we call a dehydration synthesis that join 2 monosaccharides together. Look here. If I take this hexagon and I add it to this hexagon, right? These are called monosaccharides. 1 monosaccharide, 2 monosaccharides. If I remove this OH and remove this H and dehydrate it, right, out, remove it from this molecule, dehydrate it out, I will synthesize, I will mesh together the 2 and form this molecule here, which is 1 disaccharide. 1 monosaccharide plus 1 monosaccharide makes 1 big disaccharide, right? Disaccharide because there's 2. So that was called a dehydration synthesis reaction because I dehydrated the molecule. H2O came out of these molecules and I synthesized a disaccharide. I made a 2-chain molecule. Sorry, I made a 2 monosaccharide molecule bonded together. Disaccharides could break via a hydrolysis reaction, forming 2 monosaccharides. So do you see over here that I have this disaccharide? I could introduce water right over here on this bond, right? And I could break apart this bond and the water molecule will half go here and half go here, and I will form 2 monosaccharides. This reaction, right, going in this direction, where we're going from these 2 monosaccharides to the 1 disaccharide, that happens when we build up carbs, right? Let's say we're building up a storage of them in our body. What if we need to break down the storage into smaller bits to use the fuel, right? Well, maybe we're going from this disaccharide down to the monosaccharides, okay? So this is just how do we break and make these. And what I want you to know, specifically for carbohydrates, we always called this a bond, right? We said over here that, let me go back a bit. We said that we make these bonds, right? There's all these bonds between these. So the bond, when it's sugars, right, it actually has a special name. The covalent bond that links the 2 monosaccharides together is called a glycosidic bond. Glyco, because it's this sugar, right, and this sugar together. Glyco sounds like gluco, right? So glycosidic bond because, as I said, the glyco sounds like sugar a bit, and we're forming a bond between the 2 monosaccharides, right? Now, there are 3 different disaccharides that are possible, right? So sucrose is a disaccharide. This is sucrose, and it is a glucose and a fructose that combine together, and they form this sucrose, right? And this is table sugar, right? Sucre in French is table sugar. Maltose, right? When you take a glucose and another glucose and bond them together, the red Lego block and the red Lego block, that will form a molecule. This is maltose, okay? Maltose is what is the predominant thing found in beer. It gives it its hoppiness. This is the maltose that we find, malt, right, in beer. Lactose is a combination of galactose and glucose together, right? And that will form lactose. This is a lactose molecule. That's what forms, will make up a big component of milk. I don't need you to recognize these structures and the orientation and what is a glucose, what is a fructose. What I need you to understand is if you see one hexagon, that is a monosaccharide, right? If you see two of them bound together, that's a disaccharide. If there's more than two, that's a polysaccharide. That's all I need you to know. Don't need to know these structures. I just want to prove to you that when you bond two together, they will form a disaccharide. And also that there's these three types of disaccharides, right? Because there could be a different combination of each of these, right? Actually, people that, we'll see this later on, but people that are lactose intolerant, right? This is lactose when you drink milk, right? They lack an enzyme in their body that could break this down, and it doesn't get absorbed in the body because we can't break them down into smaller bits and absorb it. And so this will actually go down into your large intestine, and little bacteria there will munch on this and make a lot of gas and upset your stomach. That's people that are lactose intolerant that get that, right? But normally we break this down. We have enzymes that can break this down. So these are disaccharides, right? And we form them. These are formed through a dehydration synthesis reaction. They are formed by building glucose and fructose together, right? And taking glucose and glucose together, right? Next is carbohydrate polymers, right? These are called polysaccharides. And so polysaccharides, these are complex sugars, right? Look at all of the hexagons together, right? There's not just one, there's not just two, there's many of them. These are many saccharides together. And polysaccharides are chains of linked glucose monomers with two main functions. The first function of polysaccharides is structural support. Some of these polysaccharides are used to build structures. So in polysaccharides in plants, some of them have this structural support, and those are called cellulose. If you look in the cells of plants, the cell walls have this cellulose. It's the most abundant carbohydrate found in the plant cell wall. Okay, so these linked together, right, in the cell wall of the plant, they make a nice thick structure. When you eat a crunchy vegetable, that crunch comes from high amounts of cellulose in the cell wall. That's the kind of protection around all of the cell. In humans, we don't really have one that's equivalent to this. Our cells don't have a cell wall. But the second part, right, is energy storage. Remember I told you in the human body, carbs, the main point to know for that is that they could be used for short-term energy, right? Well, in plants, they actually store it into these molecules that are called starch. Starch is a storage form of glucose in plants. So they will take up all of this glucose and store it. They will store it in their stems and in their leaves, right? And that storage can be broken down for there's this glucose, right? But it's stored into all of these little parts of the plant. So that's energy storage. We know it a lot more in humans, right? So in humans, we actually have glycogen. Glycogen is this branching, right, of all the glucoses. And this is how we store glucose in humans, in this glycogen. So be careful with these terms. Glycogen, glucose, it's very annoying. Glucose is a monomer, right? A monosaccharide. Glycogen is a polysaccharide, right? So when we put together many glucose in humans, right, and chain them together, that is called the glycogen molecule. That's how we store them in humans, and it's in our liver that we store this. So let's say you eat 10 plates of pasta, right? Those 10 plates of pasta, You will not be using up all of that glucose, right? All of those sugars you just ate. You'll store them, that excess sugar, in your liver for maybe later on, after you don't eat for a few hours, you'll be able to access it. So we store this glucose into big chains in our closet reserve in our liver, okay? So these are the three I care about. Cellulose, starch, and glycogen. And in the human body, right, glycogen is really the one that we care about the most. This is what how we store energy in humans. So structurally, these polysaccharides are very different. Here are the potatoes. In the potato, this is a potato cell, right? Inside of it, you see these hexagons all in a chain, right? This is trying to symbolize the starch. You find these chained together hexagons, right? This starch in plant cells are for energy storage. This is how they store their energy, right? Then in humans, right, here's the muscle from the farmer over here. In humans, you will see that there's all these, this glycogen, right? All these little hexagons linked together, and they actually branch out in this crazy snowflake type of thing. Glycogen in animal cells, they are for energy storage. We store it for our own energy use, and when we don't eat for a while, we break it down. Cellulose or fiber, right, in plants make up the cell wall. If I look at the plant's cell wall, do you see here a plant cell and you see around is the wall? I have on the outside of it, I have this thick layer, this very linear layer of cellulose or fiber. This makes up the protection of the cell. That's cellulose, okay? When we eat plants, we eat the cellulose and we eat the starch, right? So I'm going to go through each of these one by one, right? So the first one we talked about was glycogen. I said that is energy storage in humans. Glycogen is stored in two places in the human body, okay? Do you see all the little hexagons here? That's how we know that this is glycogen. This glycogen, right, is stored, one, in the liver cells. Liver cells are called hepatocytes. Cyte means cell. Hepato pertains to the liver. If you talk about hepatology or any hepatitis, right, those are like liver impacting issues. So hepato means liver, cyte means cell. So after a meal, high blood glucose, right? When we eat a big meal, we eat a lot of, we end up getting a lot of sugar, glucose, into our blood. This will cause the liver to store blood glucose as glycogen. So it'll take, we're going to feed all of our cells when we eat, but then there's so much excess. We eat like much more than we need of glucose, and the rest of it, we go and we store it as excess in the form of glycogen. So we go, all that excess glucose goes to the liver and gets stored in the form of glycogen. I'll give you an example. When you go to the store, right, you do not just buy one carrot, you don't just buy one meal and keep going back to the grocery. You buy many meals, you bring them home, and then you're going to eat the meal because you're hungry, of course, right? You're going to feed all the cells of your body with glucose. But then, right, the excess, all the other meals, you're not going to need them. Like, you're not hungry anymore, so you're going to put them in the fridge or in the cupboard, right, for storage. Well, same thing here. We eat a lot of glucose at once. We don't need all of that at once. We feed all of our cells, and all of the excess we put in storage, we put it in the fridge for later use when we're, so that we don't need to keep going back to the grocery store, right? So that we don't need to keep eating. If not, we'd have to keep eating one peanut every 2 minutes, right? That wouldn't make sense. Instead, we eat big amounts, we store the rest, and we slowly release it. So after a meal, we store blood glucose as glycogen. So we convert it into this in the liver. And when we're fasting, right, 2 hours later, when we're not eating and our blood glucose levels start dipping down, well, the liver will take its storage of glycogen and break it apart. It'll break apart these hexagons into little pieces, right? So that they could increase the blood glucose back to normal. So the liver is very good at balancing out the amount of blood sugar, okay? Skeletal muscles, after a meal, muscle cells also store excess blood glucose as glycogen. Sometimes you'll see people that go to the gym, they'll eat a lot of carbs before going to the gym, because when you go work out your muscles will take up all of these carbs, and they can build they'll help get the energy for the muscles, right? So all of these, basically, when you eat a big meal, the excess blood glucose, right, could be stored as muscles. And what's interesting is, unlike the liver, skeletal muscles are greedy. Under most conditions, muscles will use glycogen for themselves. They don't actually break it down to balance blood glucose when you're fasting. So in the day when you're not eating, your muscles will not break down the glycogen. Your liver will. Your liver is responsible for trying to maintain a homeostasis, a balance, but your muscles will not do that, okay? It's more your liver that's doing that. Only under extreme scenarios does this happen, but you really need to be in a starvation mode. So muscles use glycogen for themselves. Why are they greedy, the muscles? Because they know that they will need it. We're always moving. We're always going around. So we actually want to be greedy with the muscles and keep it for our own muscles, because we know we'll use it, right? Eventually. So that was an important part. Where is glycogen in the body? And I hope you can appreciate that it's a storage, right? It's a short-term storage of energy, because all body cells need glucose, right? But sometimes when we eat too much glucose, we store it as glycogen in our liver, in our skeletal muscle. We keep it there, right? In plants, right, the two that we saw, you're going to tell me, why are we learning about plants? Well, we need that because we eat plants, right? So we need to understand a little bit about that. So in plants, we have starch, right? Something starchy like a potato. This is a molecule for energy storage in the plant. So when plants produce more glucose via photosynthesis, they take the sunlight and they make glucose. Then they can use, so when plants produce more glucose than they store the excess in the form of a molecule that's called starch. So here is actually a little plant cell, and you see these little granules here. These granules are starch granules. So you see that they form these chains, right? This is why eating vegetables, right, is very good for us, or plants, because we get starch from them. We get this starch that they make, right? Plants also have cellulose, also known as fiber. And this is a structural support that they cause in plants, right? So on their edge of their cells, plants link glucose monomers together to form long, linear, unbranched polymer chains. Do you see how here they're branched, right? Here they're long and linear. So this is called cellulose, and it gives the crunchy of the plant cell wall. It gives that when you eat plants. Some are more crunchy than others. Some have more cellulose and fiber, things like celery, right? So this is high in fiber, and we'll talk about why that's important right over here. So, why this makes sense. This is a human anatomy class. We should talk about, how we store our sugars as glycogen. But why are we talking about plants, right? Why do we need to know about starch and cellulose? As I said, because we eat plants, right? So humans when we eat plants, we get this stuff. We get cellulose and we get starch. And humans contain enzymes. We're going to see this word a lot. These are proteins, enzymes that digest and hydrolyze, water, lysis, cut, many of the carbohydrates. Here's the deal. When we eat, right, we cannot absorb this big thing over here. This is way too big. We would not be able to absorb it. It would flush right out into our feces and out of our body. We need these things, glucose, fructose, galactose. We need small monosaccharides to be able to absorb them in our small intestine. If not, it passes straight through. So our bodies, when we eat plants, right, we eat these big things, starch and glycogen. We need to break them down. And so we have enzymes that hydrolyze, that digest many carbohydrates, right? So starch and glycogen are broken down by the human body via the enzymes amylase, released by our salivary glands and our pancreas. So our salivary glands and our pancreas have this enzyme called amylase that will break down this starch and glycogen into disaccharides, okay? Maltose, sucrose, lactose. So we start breaking things down a lot over here, right? But even disaccharides, we can't absorb these directly into our blood, right? Think of someone with lactose intolerance. That's why they get all gassy, because they don't actually absorb this. Disaccharides are then broken down by the human body via the enzymes maltase, sucrase, lactase. You're going to notice something. They all have the term ACE in them. That means they're enzymes that are cutting things up, right? So here, maltose, if you drink a lot of beer, you'll have a lot of maltose. That will get broken down by maltase into glucose and another glucose. This can be absorbed in the small intestine. Sucrose, right? If you eat a lot of sugar, then sucrase will break down that sucrose into smaller bits here, and this glucose and fructose could easily be absorbed. Lactose, if you drink milk, hopefully you have this enzyme, lactase, but my friends who have lactose intolerance do not have this enzyme, right? They will take lactose and break it down into glucose and galactose. So this is how we break down stuff in the human body. Humans do not have the enzyme to break down the polysaccharide called cellulose. Do you notice here I said starch and glycogen could be broken down? So if you eat animals, glycogen can be broken down. If you're eating plants, then the starch will be broken down. We have all the enzymes here for it. But if you eat cellulose, if you eat these vegetables high in cellulose like celery, right? No enzymes are able to break this cellulose. We do not have the enzymes to break it down because the bonds are different. So we do not have these enzymes, and therefore cellulose stays in a long chain. We cannot absorb long chains in our small intestine. So this stays in our small intestine, right? And we will just expel it out as feces. So we actually, these are called undigestible fibers. They're fibers that will not be digestible, right? This is interesting. So cellulose and fiber does not get digested nor absorbed. It doesn't get absorbed. So why is this good for us? We're celebrating this, but we can't digest this, right? That's because if you have a high fiber diet, then you will have a lot of these fibers in your intestine, in your large intestine. And actually, this is called osmosis. Water flows towards places where there's a lot of these solutes, where there's a lot of these fibers, right? So these cellulose will retain water. Water will be attracted to the high amounts of solute over here, right? And it will pull the water into this intestine, and there'll be enough water absorbed into your intestine from inside the body, and therefore you'll have good bowel movements, right? It'll be watery enough to come out and expulse. If you don't have much fiber in your diet, then water moves out of the large intestine, right? Do you see here water? There's not much cellulose. Water isn't, remember I said water is attracted to the cellulose, so not very much water will be attracted here to the cellulose. And water moves out the large intestine since there isn't high amounts of cellulose. So if there's not enough water here in the tract of this large intestine before it gets expulsed out, right, then not enough water is absorbed here, and imagine having a dry block trying to pass through your intestine. That will cause constipation, right? She does not look happy over here, and there's a sad face. So there's not much water. It's not as lubrified to get out the feces, and so this could cause constipation. So that's why pharmacy technicians should recognize that, there are these different types of products, right? So Metamucil, for example, right? That could be sold. This psyllium over here is a type of dietary fiber that could be given, right? It's kind of like cellulose. Similar to cellulose, psyllium does not get digested or absorbed. It's a capsule of things that won't be digested. It's kind of crazy we're taking that, but you could have, you could eat high amounts of fibers, or if you're really having difficulty, you could get this and it'll help you. So psyllium stays in the gastrointestinal tract and it retains water in the large intestine so that it will help the bowel movements, right? It'll help water be attracted to help flow through and move it out, right? Water reduces the friction and helps to moisten so that it'll move down the tract. All right, so that's all for carbohydrates. Now we're going to jump on to our next biomolecule, which is lipids. Lipids are an exception to the monomer-polymer system. Of course, there's always stupid exceptions. Lipids are also called hydrocarbons since they are mostly composed of carbon and hydrogen, right? Hydrocarbon. We like when names make sense. The common feature of lipids is that they have little or no affinity to water, right? This is key. This will come back so many times. Lipids have no affinity to water, and therefore we call them hydrophobic. Hydro means water. Phobic, phobia, means a fear, right? Hydrophobic means to be afraid of water. It does not want to be near water. And I'll give you an example that'll make sense. A type of lipid, right, is actually, we'll see triglycerides or fats, right? Oil is a type of fat, right? And when you put a drop of oil in water, what happens? It goes to the top and does not want to touch the water. It even goes as far away as possible, but it can't escape our glass, right? So they do not mix well, right? You even see little dots that form in the water because they don't mix well with the water. That's called hydrophobic because the lipids don't like water. I want you to now imagine a chunk of fat in a blood vessel, right? Blood is watery, full of water. If there's a chunk of fat there, right, it will stick to the wall of the blood vessel and not want to mix with the blood. And it'll clog the sides of the vessel wall, right? So what I want you to understand is when I say that there's no affinity to water, that really means that it does not want to mix well, and it'll actually try to go as far as possible as the water. So this means it's hydrophobic. Lipids in general are hydrophobic, okay? Or partially hydrophobic. So fats are hydrophobic, right? Triglycerides are another name for it. We'll talk about why that is. Triglycerides, there's 1, 2, 3 tails of fatty acids and a glycerol molecule. We'll see that in a bit. That's called a neutral fat. The fat that we have stored, right, in our body, that's triglycerides. Phospholipids are actually partially hydrophobic, but we'll get to that in a minute. I want you to know that these are the little phospholipids are what will make up the cell membrane. Around the membrane of the cell, every single cell has a border around it that's made out of these little phospholipids. And lastly, steroids, okay? Steroids are 4 rings over here, and these are also hydrophobic or partially hydrophobic. These are fat soluble. So let's look at each of these separately. Okay, we're going to go through triglycerides, then phospholipids, then steroids. So the first one is triglycerides, also known as fat. Triglycerides, let's think of their function of these fats. They are energy reserves. They could be broken down to power cells. As I said, one of the quickest ways for weight loss is, one of the ways for weight loss is to reduce the amount of food that's consumed, right? Because as you are in this fasting mode, right, you are not breaking down all of, you don't have any food to break down, so you will break down your fat reserves instead, right? So this is a reserve of energy to whenever you need it. Fat is good for you. A lot of people think that it's not good. Fat is needed. You need fat in your body to have these reserves, right? Too much of it is not good, of course. Too little of it is not good either. You need just the right amount. Fats absorb and transport fat-soluble vitamins. So vitamin A, D, E, and K, these are fat-soluble. Fat protects and cushions our organs, bones, and internal organs against shocks. If I slice someone in two in this picture, right? Do you see these little holes over here? This is the intestines, right? In between the intestines and all the visceral organs here, you see this yellow, right? You see these pockets of fat over here, right? That's called visceral fat. So you have subcutaneous fat that is in the skin, right? But the visceral fat is important because it cushions organs, bones, and internal organs against shock, right? Insulation, right? Fat under the skin retains heat. So this subcutaneous fat is very good for thermal regulation. We see in the winter, in the fall, the little squirrels start getting a bit chunkier, right? Because they start eating up to hibernate. That's because they'll be insulated from the cold thanks to their fat, right? It actually protects you and thermally gives you a layer, right? In your walls, you have insulation, you have things to prevent the cold from coming in, while the same thing in our body, right? The benefits of fats over here. So, I really like this picture. So if you zoom in onto, this is not the organs here. If I zoom into the yellow, I would see this over here. Do you see these white pockets, right? These are called adipocytes. This is fat tissue, okay? Fat tissue, all these empty little circles are actually not empty. When we look at tissues under a microscope, we put dyes to contrast the image. And the dyes bind to hydrophilic things. They bind to carbs, proteins, they bind to DNA. And so everything that's colored are carbs, proteins, DNA. But fat, remember, is hydrophobic. And our dyes that we usually use are hydrophilic. So when you see these white pockets, it's not because there's nothing there. It's because the dyes didn't bind to that, right? The water-loving dye did not bind to that area, definitely didn't, right? So this is not just air, this is full of fat. These white pockets are actually adipocytes full of fat. It's just that the dye didn't bind there. You could use other dyes and actually stain it well. This is interesting because this is where all that fat is stored. This is where triglycerides are stored. That's all that fat that you have will have these storage areas full of triglycerides. And so these are fat cells, okay? So these were the functions of fats. Next thing, the structure of triglycerides, of these fats, right? Triglycerides, if I look into this little pocket here, there will be many triglycerides, many of these in one fat cell. Triglycerides or fats are long, non-polar hydrocarbon tails of fatty acids. So don't worry too much about the long non-polar part. Just non-polar means it is hydrophobic, okay? So triglyceride fats are long non-polar hydrocarbon tails of fatty acids. Do you see these 1, 2, 3 tails? These are fatty acid tails. They are, there's an acidic part over here and there's the long chain of hydrocarbons. They are responsible for most of the fatty or the oily characteristics of lipids. So this is what makes the kind of oily aspect, the part that doesn't like water, right? So this is one fat molecule. This is a triglyceride. It has 3 fatty acids and 1 glycerol backbone over here. These neutral fats, these triglycerides, they are constructed from 3 fatty acids plus a glycerol, as I said, right? So I want to look at this one fatty acid chain over here. Do you see that there's all these C's and H's over here, right? This is a non-polar, meaning a hydrophobic tail, which is water insoluble. Water insoluble means not soluble in water. So it does not like water. It likes, these will like fat, right? So these are fats. They like to merge with fats. They do not like to be with water. This is what pushes these fats away from water, right? This side is actually polar and hydrophilic head, right? But then when it binds onto this triglyceride, it loses that ability to like water. So this whole molecule ends up not liking water. It's hydrophobic. So these fatty acid chains 3 times will add together to this glycerol backbone, right? It's this gray part here. And they will form a triglycerol. So 3 fatty acid chains plus 1 glycerol will make a triglyceride. So fatty acids could be linked together to glycerol through a dehydration synthesis reaction. They will take 1 fatty acid and bind it onto glycerol. Do you see the H and OH? That will form a water molecule. They will take another fatty acid, link it to glycerol, and make this connection here. They will take this fatty acid, link it to this piece of the glycerol, and they will form this connection. So when I go from this separate to this, that's called a dehydration synthesis reaction as I go to my triglyceride. When would this happen in the human body? When you're building up fat, right? When you are making reserves of fat, maybe you just ate a lot of fat or you're storing, you need to go and store things, you will build up fat like this, right? Maybe after a little bag of chips. Then when you go to the gym and you start running, you will get rid of all your glucose in your body, your glycogen storages, and then, right, you will start to burn fat down. So you will start taking this triglyceride and hydrolyzing, water, lysis, break. The water will enter into these bonds here and break apart the bonds and separate them so that you could use them for energy. You will be able to convert them into energy later on. So That's hydrolysis to break down if you're going in this direction, right? So fatty acids could be linked to glycerol through dehydration synthesis reactions, and they could be broken down through hydrolysis reactions, just like carbs. So that was all for triglycerides. These are the fats we think about when we talk about lipids. We know about these, triglycerides. This is the fat we always talk about. Phospholipids, we don't know much about. In general, we don't talk about this very much on our day-to-days. These phospholipids, the function of them is that they are the major constituents of cell membranes. If you look at a cell, it's a sphere or a weird shape. The outer part of it is called the cell membrane or the plasma membrane. That cell membrane is made up of this double layer of phospholipids. Do you see the head and the two tails? This was one head of glycerol and 3 fatty acid tails. This is one head and 2 fatty acid tails, right? That makes up the little phospholipid membrane. It's a double layer of it. And the structure of the phospholipids is that there is this head over here and the 2 tails. The head is made up of a phosphate molecule. Do you see the P over here? That's a phosphate. There is a glycerol. All of this is a glycerol over here. That's your glycerol. So the phosphate is what's different. That's why it's called a phospholipid. And then the tails over here, do you see these tails? These are 2 fatty acid molecules that are just sticking out, right? It's not 3 like the triglyceride. So that's how a phospholipid looks. And I want you to know, we're going to talk about this in a minute, the tails here, they're still hydrophobic. Remember I told you over here that the tails, these tails were hydrophobic, right? Well, this head, however, right, this is going to be hydrophilic. Do you see these little negative charges and positive charges in here? This is going to be hydrophilic. This is going to be water-loving, and this is going to be water-fearing. Okay, so the tails are water-fearing, and the head is water-loving. So we're going to see over here, this is called amphipathic. We have hydrophobic things, hydrophilic things, and we have things that are a bit of both, right? Philic means to like, phobic means to fear. Phospholipids are amphipathic. They are amphi means both, right? Amphipathic means that they have a bit of hydrophilic parts and a bit of hydrophobic parts. So the head here is a hydrophilic region, and the tail here is a hydrophobic region, which is very interesting because when they assemble together, right? I want to go over here. When they assemble together, when you throw them in water, they actually come up with this weird format where the heads will face the water on the outside. These heads will face the water on the outside, and the tails flip inwards to point towards themselves. This is one phospholipid and this is another phospholipid. They are forming this double layer of phospholipids when they merge together. And that's because, this is why we call it a bilayer, because when they're in water, phospholipids will self-assemble into the bilayer and shield themselves from the hydrophobic portions from contacting water. And this will make this bilayer, right? Think of inside of a cell. You're going to have full of water. Outside of a cell, there'll be a lot of water as well, right? So what these phospholipids do is they put all of their heads on the outside, which will go in over here, will access the water on the outside of the cell. These heads love water. And on the inside, where there'll also be water inside the cell, these heads on the inside, they will touch water. But the tails, who hate water, right, they will go and face each other to hide themselves from the outer water and inner water, and that's why they form these bilayers and form these spheres, right? So it's really interesting how they place themselves. If I took just one phospholipid and threw it into a pool, you'd have the head that would be touching the water and the two legs would be pointing out, right? Because the tails don't want to touch water. But if I take a bunch of these phospholipids and throw them in, they will arrange in this orientation so that everybody's happy. The heads are all touching water, but the tails are protected on the inside and they don't touch water, right? So, what are the structural differences and similarities between phospholipids and triglycerides? I'll pause and take a minute to let you take a look at this. All right, so the triglycerides, right? Triglycerides. They will have 3 fatty acid tails, right? 3 fatty acids. They will have 1 glycerol. And all of this together, right? Remember we said that all of this is hydro water. Phobic or philic? This will be phobic, right? Because it has a phobia of water. It does not like water. Altogether, this molecule doesn't like water. You know this. When you see fat in a glass, right, it floats at the top and it kind of does not want to touch the water, like oil, right? And that's the structural differences, right? Then the phospholipid, these, right, will have they will have part of a glycerol also, right? They have, what's different is they have a little phosphate group, right? So this is different. And they only have 2 fatty acid tails, right? And this part is hydrophilic, right? Hydrophilic. This loves water. Philic. And this part down here, the tails are phobic, right? Hydrophobic. They don't like water. Hydrophobia. So altogether, this is called amphipathic. They half of it likes water, half of it doesn't like water. So that's the general structural difference. I know it's not in the question, but I'll ask you now, what are the functional differences, right? What do they do in terms of functions? Triglycerides, these are the fats of the body. These are for long-term energy storage and protection and all of these things. Phospholipids don't do that. Phospholipids are around every single cell. Every single cell has some fat, phospholipids. Sorry, not fat. Every single cell has phospholipids that line the outside of it and make the protective border, right, of the cell. Without a phospholipid layer, there wouldn't be a cell. Like a house, you need to build walls around a house. Like that is the same situation for cells, okay? Awesome. Now steroids. Steroids look a bit different, right? Don't confuse this for a polysaccharide. I know I told you to look for hexagons, but if there are 4 fused rings together, 1, 2, 3, 4 fused together, that is called a sterol, right? And that's what makes up the backbone of a steroid, okay? These sterols form the backbone of a steroid. Some examples of some steroids are cholesterol, right? Sterol. These are components of the plasma membrane. In between the little phospholipids, you find these cholesterols, and they help with membrane fluidity. They help to make the membrane more rigid and tough, or more loose and allow things to go through a bit more. So they help to maintain the stiffness of the membrane. The bile salts, right, are produced. These, do you see 1, 2, 3, 4? 1, 2, 3, 4. These are sterols. These are still steroids, right? Bile salts, they are produced by the liver. They are stored in the bile. And then they get released into the small intestine, and they help to break down fats. To break down fats, you need something that is fatty and hydrophobic, right? In chemistry, we say like dissolves like. If you just washed dishes with water, it wouldn't do much to the oil and the grease that's on the plates. To break down that fat, you need soap, which is hydrophobic, that will break down all of, which will wash away all of that fat and be able to dissolve it off of the plate. Same thing with salts. To break down the fats that are in the small intestine, they use bile salts that are able to emulsify the fats and break them down into smaller bits. Hormones, there are some steroid hormones, and these are particularly the sex hormones. There are others, but these are the important ones. So testosterone and estrogen are the sex hormones that we think about. There's progesterone too. And these sex hormones are steroids as well, right? Taking steroids, right? That is thought of as sometimes some of these hormones over here. So that's the general idea for the steroid. I just want you to keep a note, like for these lipids, triglycerides, phospholipids, and steroids, all of them have a common theme that you need to remember, is that they are either all or partially hydrophobic. All the other biomolecules we're going to see. So we saw carbs, we're going to see proteins, nucleic acids, all those other ones, right? They are all hydrophilic. They love water. They swim in the water, in the blood, in the body. They go around. But these lipids, they're picky. They have parts of them or all of them that are hydrophobic. And that's what makes them this category. If you want one general note about lipids, it's that parts of them or all of it is hydrophobic, doesn't like water, right? So that's particular because our body is almost all has a lot of water in it, right? So they have to be used in different ways, and as you see, their properties make some pretty interesting things in the human body. So pause the video. Here's a question. I want you to identify. These are all lipids. I want you to identify what they are. Okay. So pause the video and take your time. Over here, right, we see some hexagons. Do not tell me that this is a carbohydrate, right? This is not polysaccharide. This is 4 fused rings, so we know that this is a steroid, right? This over here also is a steroid. It has 1, 2, 3, 4 fused rings, okay? This over here, these crazy letters, I see 1 tail over here, 2 tails. This is the fatty acids, right? This is hydrophobic, this area. This over here looks like a glycerol, and this looks like, I see a phosphate here. Do you see the P? So this must be a phospholipid, right? This must be a phospholipid. The thing that really pushed out to my eyes was the 2 tails, right? Oh, that's a phospholipid. Here I have a glycerol, right? You don't need to recognize that. What really made me think about this was the 3 tails that are here. 3 tails, this must be a triglyceride, right? Triglyceride. also known as fat, right? Fat. So that's how we identified those. Next one, we did carbs, we've done lipids, now we're on to proteins. What are proteins? Aha, a little bit different. Proteins are made up of carbon, hydrogen, oxygen, and nitrogen. Proteins, also kind of known as polypeptides, are made up of polymers, polymers made of amino acid monomers. Do you see these amino acids that I'm just showing as little circles over here, right? These monomers of these amino acid monomers, they come together and form a chain that's called a polypeptide or a protein, okay? So in this polypeptide, I'm going to notice here that there's going to be one end with a nitrogen and there's another end with a carbon, okay? We'll see all of that later. And these are peptide bonds in between. That's why it's called polypeptide, many peptide bonds. We're going to see that in a bit. But just, okay, so remember in carbs, there was monosaccharides making polysaccharides. Now it's amino acids forming proteins. I know this name is weird, but I'm going to tell you why they're called amino acids just now, okay? So protein-related terms we should be okay with is that amino acids are a single protein unit or monomer. Do you see these little dots? These are all amino acids that are separate. We link them together to form little peptides. This is 2 to 50 linked amino acids. More than 50 linked amino acids, right, are called polypeptides. And when you take that polypeptide and you fold it up in the correct orientation, this will be called a protein. A protein is a functioning polypeptide that's all folded. To function properly, proteins need to be folded correctly, okay? So this is going to be key later on. A lot of people will mistaken polypeptide and protein. Polypeptide is just the linked amino acids, right? When you fold it up correctly and it can function, then you can call it a protein, okay? Sometimes many polypeptides even come together to form a functional protein, right? Sometimes you need a few separate polypeptides to come together. So we'll talk about this a bit more in a second. But I want you to be aware of these terms. The term peptide also sometimes is referred to as the bonds between the amino acids. So we'll talk about that nuance too, but these are kind of the key takeaways, right? Remember in carbs, we saw monosaccharide, disaccharide, polysaccharide. Well, here we have amino acids that are 1, like monosaccharides. We have peptides that are 2 to 50, kind of like the disaccharides, right? And we have polypeptides, which are like polysaccharides, right? So we're going to go through each of them one at a time. Amino acids, these are the protein monomers. There exist 20 different amino acids which all contain the same thing. They have a common component. So this is one amino acid. Do you see these little dots here? One of these dots, if I looked into the chemistry of it, would be all of this, okay? And that, so one of these tiny amino acids looks like this. They have a common carbon in the middle, okay? Do you see the carbon that's right over there? So here's my common carbon in the middle, right? Then I have an amino group on this side that could either be written as NH2 or NH3. If you took some chemistry classes, you'll remember ammonium, right, has this N inside of it, right? So this over here is the amino group, ammonia group, right? This is the N side. On the other side, you will have the carboxyl group. because there is a carbon and an oxyl group, right? Oxygens will be there. So COOH or COO minus. This is going to be the two options that you might see. This is the carboxyl group that we see here. So there's an amino group, a carboxyl group that are linked to the central carbon, and there's a hydrogen group that's linked to the central carbon. That will always be there. Every amino acid, the blue one, the green one, the pink one, the yellow one here, they will all have an amino group, all have a hydrogen, all have a carboxyl group, but the different amino acids will all have a unique R group. This is called the side chain, and that will determine the chemistry of the amino acid. There's not this is not the element R, okay? I wish they called it the question mark group. So they will have a unique something over here in the position of the R, okay? So it's not R for real. I don't know why they call it R. I should look that up. This will be something else, right? So basically what I want you to know, all the amino acids in the world have this section, but the R group will change. So some chain over here will come in the R place. And you do not need to memorize these. I want to make you understand the concept. What they are simply showing here is that there are 20 different types of amino acids. But as I told you, there's always the common component. Remember the amino, the carbon, the hydrogen, and the carboxyl. There's always that. Look at the purple part here. You see the central carbon, right? Amino group, hydrogen, carboxyl. Amino, carboxyl, hydrogen, central carbon. It's all the same. They're copy pasted, right? Do you see copy paste, copy paste, copy paste? All the purples, they're always there. That's this part here. That's why I said it's the common components. Central carbon, amino group, carboxyl group, hydrogen group, always repeated, right? Then you have many different side chains that are called R groups. Remember this R group here? It's not actually an R. It's all the colorful stuff, right? So this amino acid called glycine has a hydrogen as its R group. This amino acid called alanine has CH3 methane as its methyl group, right? This amino acid is married and has a little ring over here of carbons. This is phenylalanine over here. There are many different types of, there are 20 specifically different types of amino acids, right? And they all have these different types of layouts, right? Okay, so it's interesting because last thing to say is that these R groups that are very different, they all have different properties, right? Some of them will be hydrophobic side chains, right? Which means that they won't like water as much. Some of them will be hydrophilics, and some of them will be charged with these low negatives and positives. So it's interesting. They all have different functions in a protein. So that's that. Now, if I look at, we shouldn't be scared of these, right? This is one amino acid. This is another one. There's the amino group. There's the carboxyl group. There's the hydrogen, and there's the R group. I'm going to go back 2 seconds. One thing I forgot to mention. Amino acids. Look at the name of it. It's an amino, right? This is the ammonium side. Acid, because this is a carboxyl group, and sometimes in chemistry we call this carboxylic acid over here. So this is why we call it an amino acid, right? This is why the one little dot here this little dot is called an amino acid, right? Because each of them have the amino group and the acid group, the carboxylic acid group, okay? Forgot to mention that. So here's one amino group. Here's one amino acid. Here's another amino acid. And we're going to talk about making polymers or breaking them, right? So peptide bonds are going to be formed to make a protein polymer. We're going to see this in a minute. To form a polymer, right, we need to have a dehydration synthesis reaction. So we need to dehydrate, remove a water from these two amino acids, right? Remove the water, and we synthesize a bigger now dipeptide, a protein, right? So in this case, right, this dehydration synthesis reaction occurs between the amino group of one of these amino acids. So, sorry, here's the amino group of this amino acid. Do you see how there's a hydrogen here? That'll be gone. And it occurs between the carboxyl group of another amino acid. So this OH over here will be removed. So this OH gets removed, this H bonds and forms a water molecule over here. And that will form a bond, a peptide bond between these two amino acids over here. So amino acid 1 plus amino acid 2 forms this dipeptide over here, right? So bonds linking adjacent amino acids together, these are called peptide bonds, right? When you form a link between one amino acid and another, it's called a peptide bond. Remember in our section on disaccharides, right? We saw that one monosaccharide and another monosaccharide came together and it formed a glycosidic bond, right? Well, when an amino acid and another amino acid come together, they form a peptide bond, right? So between two amino acids, 1 and 2. So I like this little layout because here you have the backbone of one amino acid and the backbone of another one, and you have the R group over here of one of them, the R group of the other, and you see that these are linked together. This is called a dipeptide because there's two amino acids linked by a peptide bond here, right? You have the central carbon, you have the nitrogen group, you have the acid group, you have the H, and you have the R group. Same thing here. You have the COOH, you have the acid group, you have the nitrogen group, the hydrogen, and the R group. And now we're going to add this amino acid and make a tripeptide with 3 different amino acids, and this OH of one amino acid and this H of the other amino acid will merge together, remove the water, dehydration synthesis, and it'll form a nice little peptide bond here, right? And you see that all the side chains are over here, and you see all the backbones, and you see 2 peptide bonds, right? So let's take a look at this question. Pause the video and fill out this question over here. Alrighty. How many amino acids are present in the peptide below? So what I want to do first is circle some important things. I see on this side the amino group, right? I see over here another N that got merged together, right? I see over here another N that got merged together, right? So that's your amino groups. Then the carboxyl group. I see the carboxyl group over here at the end, right? I see the carboxyl group over here at the end, and I see another carboxyl group over here, right? So I now want to go in and have my central carbons, right? This is my central carbon, I guess. This is my central carbon, right? This is my central carbon. Remember, the central carbon is always going to be sandwiched between an amino group and a carboxyl group. So over here, central carbon, amino, carboxyl. Central carbon, amino, carboxyl. Central carbon, amino, carboxyl. And now I'm going to go and get my hydrogens, right? Remember, all of them have these hydrogens. And now I will go and circle the R group. Here's my R group. Here's my R group. And here's my R group, right? So how many amino acids do we have? Well, you could just count the number of central carbons. 1, 2, 3, right? So the answer here would be 3. Draw an arrow under the peptide bonds over here. Well, we know that peptide bonds occur when you merge together the amino group of one and the carboxyl of the other, and you see between each of these is a C double bond O and an NH. C double bond O, NH, C. So you know that that's what's to look for, right? So over here is one of the peptide bonds between the C double bonded O and the N. That's where they bonded together. And then over here is the other peptide, right? So there are 2 of these peptide bonds for 3 amino acids, right? Circle the R groups. I did that already. That's going to be in green. If you look at this, you'll see it in this green color over here. Okay? So, I think we're good now. Protein, this is an important concept. Protein folding is crucial, very crucial. Proteins have a hierarchy of structure, and they're organized into 4 levels. I want you to just go look at the names here. So glycine, alanine, valine, leucine, right? These are all separate amino acids, but when we draw pictures, we often draw circles. But you know that every amino acid looks like this, right? So glycine, proline, threonine, blah, 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 right? This is a whole chain of these together, a chain kind of like this that we just saw. This is a chain of 3. This is a chain of 1, 2, 3, about 20, right? This is called the primary structure. The polypeptide's unique sequence of amino acids is called the primary structure. What is the order, right? Here I could have put proline after threonine, and that would have changed the protein, right? Or I could have put methionine in the place of lysine and swap them, and that would have changed the protein. So the primary structure is the order of the different amino acids, right? You could have a random order. Think of it. There are 20 different amino acids. The combinations are endless of the chains of these that you can make. Then, right, if I look at just this section here, these 3 amino acids, there could be different types of organizations of folding. So this little amino acid here, right, with this grouping will maybe make these sheets, right, all folded. So this portion of the chain, the amino acids that are chosen there, right, they could fold and coil on themselves in different ways. So this is between the backbone components. So between not the R groups, right, but between the actual backbone, the carbon, over here, like this purple section, right? So between the amino groups and the carboxyl group and the hydrogen and the carbon here, that will cause different secondary structures. Maybe they'll interact in a certain way. And there are really 2 that we care about. There's these beta pleated sheets that form these foldings like this. Or sometimes, right, there are these alpha helixes. Sometimes the way that the different amino acids are in order will form more of these ribbon type of interactions. Do you see this backbone piece interacting with this one? You see these little dots here? That's showing like an attraction. That, because of the different order, this could change the folding of the secondary structure. And then once we look at the bigger picture, right, the overall 3D shape, the conformational shape of the polypeptide, right? That will also result from interactions between the R groups, right? So do you see the little folded sheets here? That's the beta sheets. That's like what I call local folding. This is a little local folding in terms of a helix, but the overall folding of all of this into a ball, right? This little potato shape happens because the R groups interact with each other and form the tertiary structure. And then sometimes, not always, some proteins have a quaternary structure. Some proteins have many polypeptide chains that come together. This is one polypeptide that was made, and it formed this shape. But maybe 4 separate polypeptides that were made come together to interact and form this quaternary structure, right? Many polypeptides could come together. Sometimes it's 2, 3, 4, 5, right? They can form this quaternary structure that is many polypeptides together. So I think what people miss in this diagram is that this is showing the same exact thing. This over here is actually this tertiary structure. This tertiary structure includes many different secondary structures, right? And in that secondary structure, the reason it looks like that is because we have this primary structure of different amino acids. So this is just zooming into the depth of each of these hierarchies of the polypeptide. So I want to go through these one at a time. The primary protein structure is like the orders of letters in a long word. So amino acid sequences will determine the primary structure and will thus determine its function. So here, there's a bunch of abbreviations, right? But this has glycine, proline, threonine, right? There is a order of all of these over here. This protein has 127 maybe amino acids, right? So each of these, this glycine is here. This proline is here. This threonine is over here, and on and on and on, right? So this is one specific protein that has this order. Another protein has a different order. Just like how one book has an order of words, a separate book that you read will have a separate order of words that means something totally different, right? And that will affect its function, structure and function. Remember, that's important. The secondary protein structure, right, we said was over here. That results from weak bonds called hydrogen bonds between the carboxyl and the amino groups. And this will cause the protein to fold into patterns. We said there was 2, right? Sometimes there's little weak bonds, hydrogen bonds, that will form an alpha helix, right? Sometimes there are weak bonds between the sheets that will form these beta pleated sheets in some sections. These are called local foldings. In some little regions, there's sometimes some folding that happens in a helix way or in a beta sheet way, okay? The tertiary protein structure refers to the overall 3D shape and therefore the overall function of a polypeptide, right? Do you see here how we had some sheets here, some alpha helices here, some sheets here, some alpha helices, but the overall folding, right? Like this section is close to here, and then this section folds in this way. All of that forms the overall 3D shape, and therefore the overall 3D function, right? And so this is due to R groups interacting together. And lastly, like I said, quaternary protein structure. Not all proteins have a quaternary structure, but if a protein is made up of multiple polypeptide chains, then a quaternary structure does exist, right? So for example, here, so this protein, for example, is one polypeptide. If you follow the chain, right, it's one long strand of amino acids. And So this is gonna not have a quaternary structure. But this protein here, right, has 1, 2, 3, 4 separate chains, right? So a hemoglobin has four polypeptide subunits, and they're joined together through weak bonds. And this arrangement facilitates oxygen transport in the blood, right? Because you could carry 4 oxygens at once. Collagen is made up of 3 coiled polypeptides, 1, 2, and 3, right? And they join together and they form this tight rope together, right? They're separate chains, but they form a tight rope. And these create a strong protein fiber that's found in a lot of connective tissue, or collagen for your hair, strong hair, right? So that's the quaternary structure, is bringing together multiple polypeptides to form a functional protein. And so remember what I said, that protein structure equals function. Structure, how the protein looks, means what it's going to do as a function. Remember I told you, if I give you a hammer, right? I don't think you're going to do a good job as a teacher. If I give you a hammer. If I give you a computer with access to PowerPoint, then maybe you will be a good teacher because you could design things, right? If I give the construction worker a laptop, not very useful to use on the job, right? So structure equals function. What you have as tools, what you look like as a protein, will tell me about what you're going to do as a job in the cell. So the folding of a protein is an important process. A functional protein consists of 1 or more polypeptides that are folded into a unique shape, okay? So the sequence of amino acids will determine the protein's 3-dimensional shape. Remember I told you here, if I change one of these, that might change the helix, or it might change the 3D shape, and that might change how it interacts and does its function, right? The shape, the hammer versus the laptop, right? That will change its function. So it's important to have the correct amino acid sequence if you're building a protein, and that's the protein that'll do this job, right? So for example, this is an antibody protein that has a certain shape. This antibody was made and has this shape to destroy the protein from the flu virus. This is the flu virus. Do you see its shape, right? The shape of this specific protein on the flu virus. This attacker antibody that we build in our body will fit so perfectly onto this piece of the flu virus because structure determines function. And I will not shut up about this. Structure determines function. What it looks like will tell you a lot about its job. This protein needs to bind here to be able to destroy this virus. So that's very important that our proteins are folded well. That's what my key takeaway is. You need proper folding. You need the proper sequence of amino acids for it to fold, because if not, it might not be able to do its job well. I'll give you an example of this. So if we alter the primary structure of a protein, we can cause differences in the structure of the protein and therefore its function. Here, the primary structure of this protein, this is hemoglobin, okay? And hemoglobin carries oxygen, and the primary structure is in the beginning is valine, histine, leucine, threonine, proline, blah, 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 right? So this is the normal primary structure. It folds up and forms this secondary and tertiary structure that looks like this, a glob. This is a cartoon, right? But it's like a glob shape. This is a normal beta subunit of the hemoglobin. And actually, when you have this little polypeptide here, it binds to 4 other polypeptides and forms the hemoglobin. Remember over here, I told you hemoglobin is 4 polypeptides. Well, over here, you see that there's those 4. And proteins do not associate with one another. They're separate, right? And they do their job. Each of these carries 4 oxygens, and all of these are in the red blood cell, and they pick up oxygen from outside and they deliver it to places. They're found in this red blood cell, and they look very nice like this. In a condition called sickle cell disease, there is one amino acid number 6, right? Converted from this amino acid to valine over here. And the subunit, right? The normal subunit has a little sickle in it, right? It has this little dent in it. Just because this valine, remember that the valine has an R group. It has a different R group compared to this glutamine over here. So this will have a different shape because of just one amino acid. And that will cause the hemoglobin to look like this. The beta chains will have this divot. And weirdly enough, because of this divot in this disease, the proteins end up aggregating and they are attracted to binding to these little parts over here, and they form actually these long chains. And that really decreases the ability for the hemoglobin to carry oxygen because they bind like this. Do you see here that the proteins don't bind like that? But because of this divot, they kind of, like a Lego set, they match up and they form these big structures here. And this is what our red blood cells look like. So this is a sickling, a half moon, right, of the red blood cell because these long chains are inside. And the hemoglobin don't carry as much oxygen as they could. Do you see here? This is an example of how in sickle cell disease, one amino acid that went wrong affected the secondary structure and the tertiary structure, which affects the quaternary structure, which affects the functioning of the protein, and therefore the protein cannot carry as much oxygen, right? Maybe because it can't access this area over here, right? So structure equals function. That is key. I will repeat it over and over and over again. So the folding of a protein, this is not well folded, right? It's essential. What is denatured protein? Is a denatured protein. Proteins require a specific 3D structure to function properly. I will keep saying this over and over. If that 3D structure is disrupted, this is called denaturation, right? You're naturally like this. If you denature it, you destroy its normal nature. If you denature it and disrupt its 3D structure, the protein becomes non-functional. It cannot work. So over here you have a normal protein that has this shape because of the amino acids in it. And if you denature it, unfold it into the spaghetti form, right? This is the nice compact form. If you unfold it into spaghetti form, then it's denatured, okay? And 3 factors can denature proteins, right? So extreme temperature can change proteins. High heat or different freeze and thaw cycles are changes between temperatures can denature a protein. This is important because, our bodies are full of proteins that do very important functions, right? We get scared when our internal body temperature increases. This is called fever, right? If our body temperature gets too high, we are told to rush to the hospital, right? Because this is not good for your body, right? We do not want our proteins, the workers of the cell, to denature. That would be awful. Proteins keep us alive. They do the work in our cells. We cannot have them denaturing and not functioning, right? freeze thaw cycles are like taking things out the freezer, putting them, unthawing them, putting them back in the freezer, unthawing, right? So this is bad too because it's extremes of temperatures. We'll talk about that in a second. What else can denature a protein? Can unnature it, right? Changes in pH, so strong acids or bases, right, can change our proteins. Changes in salts, right, concentrations can also denature. So in our body, in our blood, we want very stable temperature. We want very stable pH. We want very stable ions and salt concentrations, right? Some medications that we take, this is applicable to us, they are protein-based, right? An example of this is insulin. Insulin is a protein-based medication, and it must be treated with care to avoid denaturation, right? So when you handle, there's a reason why certain meds, you need to follow the protocol. Protein medications are not taken orally, for instance, because stomach acids would denature them. That's why we inject it. It needs to go directly in the bloodstream because if it were to pass in our stomach, it would get denatured and broken down, right? So we actually, protein meds, we need to inject them. Protein medications must be stored at the correct temperature, right? So we need to keep protein meds at a right temperature and avoid them from freezing them, thawing them, freezing them, because that's not good for them. They will denature. So we need to follow the protocol for whatever that med has as a temperature, right? Protein medications must be mixed and reconstituted with the proper recommended ion solutions to keep them in the right salt concentration, because if not, they will denature, right? So when it says to put it in this much saline salt, right, we need to make sure we follow that protocol and not mess up our concentrations. Protein medicines are typically enteric coated to avoid denaturing. So protein-based medicine is administered orally, any protein medicine that's administered orally must be what we call enterically coated. Enteric sounds like digestive processes, stomach, breaking things down. So enterically coated means to have a little coat around the drug, right, around the medicine. Because if this protective layer was not there, the very acidic pH of the stomach would quickly denature the drug that is protein-based, and it would render it dysfunctional before it even had the chance to be absorbed in the intestine, okay? So here's my drug. Here's the enteric coating, right? The drug stays intact in an acidic environment of the stomach, right? So when it's enteric coated, right, we take it in orally and it has this coat. In the stomach, it stays intact. And then once it gets to the colon, right, the intestine, enteric coating is solubilized by colon bacteria, and drug matrix is swallowed up. So over here, all of this coating here will get broken down once the colon bacteria have seen it. And then the drug is going to be able to be there, and the drug could be released into the digestive system and it could be absorbed, right? So this is a smart way to give protein-based meds, and we understand why it needs to be coated. There's no point of taking this protein drug orally if we end up breaking it down in our stomach and making it useless, right? The other way we could deal with this is just injections into the blood directly, correct? So that's how we deal with protein-based meds. Proteins, I said, are the workers of the cell, and they have so many functions. Just like in a little city, a little cell, you have people that do different things, proteins that do different things. So we're going to see all 7 of these, okay, one by one. The first one is structural support. Here you have, so for an example of this support is keratin. It's a protein in the hair. And basically, right, this is this collagen over here. This is a type of protein, right? So this collagen helps to kind of have this elasticy property and strong structure. So this provides a good fibrous framework in animals' connective tissue. This is the tissue that's right under the first skin layer, and it forms this meshwork. I call it the superglue. You see these big fibers that form this nice superglue mesh network. That's structural support in our body or in our hair, right? Our skin or our hair. So that's collagen. This is just an example. You don't need to know the details of this, but an example of a way that proteins structure the cells, right? Or the tissues. Storage. So a lot of proteins will be used for storage of amino acid. A specific protein in milk is a major source of amino acids for the baby mammals, right? Also, plants have storage of proteins in their seeds. Ovalbumin is a protein of the egg white that's used for amino acid sources in the developing embryo. So all of these are some examples of storage proteins. We store even some, our muscles have a lot of these amino acids stored as long proteins there. If in a very dire scenario, we can break down our muscles, right? But we really want to save that till the very last resort. Transport, right? We transport substances through. Remember, I told you proteins are also the bridges of the cells. This is the outside world of the cell, right, in the human body, and this is inside the cell. And it can transport things into the cell or block other things to not come in. So there are many different proteins, transporters, that let things across, okay? Because sometimes they can't pass through the cell membrane. Remember, the cell membrane has a hydrophobic area. So things that are hydrophilic, like proteins, or carbs, right? They will need a little bridge to get over. Movement. Some proteins are contractile motor proteins. Actin is one protein. Myosin is another protein. And they will move, right, inwards and outwards. And they will contract to like your muscles, right, that contract to allow you to move. Some proteins are used for cellular communication. So I gave you the example before of a hormonal protein. That's a cellular communication because when you have high blood sugar, insulin is a protein hormone that will cause the blood sugar to go down. So hormones are proteins. Receptors. What is a receptor? Okay, this we'll talk a lot about later on, but a receptor, right, is a response of a cell to a chemical stimuli. So here's my cell in orange here, and here's a little purple receptor, right? And a neuron here is releasing signals onto this cell to tell it to do something. Well, the little receptor is like an antenna that picks up all the signals. So here you have the signal picked up over here, and the receptor will then tell the cell what it's doing, right? So I like to think of it like a little, it's like a phone, right? When someone calls you, you're receiving some information from them, right? You could do something in your own house if they tell you to do something, right? So that's communication in general. We have defense. So some proteins are called antibodies, and they are little attackers that will bind to viruses and destroy them, right? Or bind to bacteria and destroy them. We'll go through that in the immune system. And some proteins speed up reactions. So some proteins are called enzymes. If all goes wrong, this is a function I need you to know about. Enzymes are important. and we're going to talk about them. Enzymes, their names usually end with ACE. We saw some already. We saw lactase, sucrase, maltase, amylase, right? Anything with an ACE is an enzyme, and these speed up chemical reactions. So an example of this is an enzyme, maybe it's lactase, and it breaks down this lactose molecule into two separate pieces. If this were left in the body, this would only break down after many years, right? You need scissors in the body to cut things up and build things. That's enzymes. Enzymes, they will help, for instance, hydrolyze and break food molecules. They could also help build things. So when you build something in your cell, they can piece together things quickly. It's like a pocket for things to happen in. So let me talk about enzymes. Enzymes are proteins that speed up chemical reactions. So here's a little enzyme over here, and they build up certain things, right? They help to speed up things that take milliseconds instead of years. So they help to break or make. Enzymes often end in ACE, right? And they are responsible for cellular metabolism. So they help speed up our reactions, right? To have a cell metabolism, to have things go on inside the body. Here's an example of an enzyme, okay? So enzymes on the surface of our intestinal cell, they break down sucrose, which is a disaccharide, into glucose and fructose monomers. So here's sucrose. This is a disaccharide, and sucrose will bind onto sucrase, and it'll form this little complex. And now this protein makes it easier to break the bond here and form glucose and fructose, right? Let's look at this little animation here. Here's the enzyme, right? You have your sucrose that lands in here, that binds in the enzyme, and it easily splits apart the two. There's a little chopper here that will help to break the two down. So that's what an enzyme is. The enzyme substrate complex. So a substrate, right? This little substrate here will bind to an enzyme, the red enzyme, at the active site, we call it. And it'll form what we call the enzyme substrate complex. They're complexed together, the enzyme and the substrate. And the active site, right? This is the little active site over here, is a specific region of the enzyme that binds a specific substrate, right? This little teardrop fits nicely just because the active site is round like this. If it was square, it wouldn't fit, right? So products, when they bind together, then boop, something happens, right? There's a chemical reaction that happens, and products are released from the active site. But I want you to understand that enzymes always remain unchanged, right? They always stay as is in the body, and they do their job of chopping things or making things, right? So that's the enzyme substrate complex. Environmental factors affecting enzymes. Enzymes work best under certain conditions. I told you, enzymes are proteins. Proteins can denature. How can they denature? If there are unfavorable temperatures or pH, right, that can denature the protein. Do you see this nice protein in green? This is an enzyme. It's binding this yellow substrate. If there's denaturation, heat or pH changes, that could denature the protein. And now, all of a sudden, this yellow little substrate doesn't fit into here anymore, right? That's called denaturation, denaturing the enzyme. Enzymes are proteins, so this happens to them too. And this is important because that renders the enzyme non-functional, right? This can't do its job anymore. Again, as I said, in a patient that has fever, let's say, If the fever is too high, we start freaking out at the hospital because we need to decrease the body temperature. They will stick people in a cold bath just to decrease their temperature if needed. It's not ideal, but this is important because this denaturation, right? We do not want enzymes to break down. They are doing essential things in each of our cells. So what is enzyme activation energy? So this is called the EA, the activation energy. It's the difference in energy between the reactants and the transition state, an intermediate step where bonds are broken apart. So let's take a look at this. This is the energy of molecules. Do you see this molecule here? Two little blocks, two monomers, right? So this is the reactants, right? And it has this much energy, right? Because there's a bond here with a lot of connections together. This will form these lower energy products, right? So we need, these things over here have a bond together. This has a lot of energy. When you have a strong bond with a friend, there's a lot of energy between you two that you can give to each other. So cheesy. When you break that bond, you have this lower energy products. That bond is no longer there, right? Very sad. You have these separate products and there's no bond between the two. You actually release that attractive bond between the two chemicals, right? So this has lower energy. So to get from here to here, we need to put in some energy to unstabilize the bond, right? So the EA is actually this minimum amount of energy that is required to start a chemical reaction, like breaking something down, and then it goes down, right? Like then we can go and have our little products here. So usually you have some energy you need to put in to break the bonds, and then you make a lot of energy when you break the bonds, right? But the EA is the intermediate step, right? The activation energy to be able to break the bonds, you need to put a bit of energy in it, right? So you need a little bit of an ignition, right? An example of this is over here, like to roll the rock down, right? You need to put in a little bit of work to bring it up to the top, and then it'll have enough energy to roll all the way down because you're increasing its potential energy, right? So how do enzymes speed up reactions? They actually lower the EA, the activation energy. Enzymes catalyze chemical reactions by decreasing their activation energy barrier, so that chemical reactions occur a lot faster than they normally would. So how does this happen? Here's our reactants, here's our products. Normally, without the enzyme, you have this peak, you put in this much energy, and then it will break apart. For example, right? To light up a fire, you could put the wood, you could put the paper. There's a lot of energy in that wood, in that paper. But you need to give it a spark so that it has this transition state to begin breaking it down, right? And then you could burn and break down the wood and burn and break down the paper. So you need to put a bit of fire, ignition, right? And you need oxygen to reach the transition state, and then it will break it down, right? And then you can release energy. What enzymes do is they will decrease the activation energy, okay? So normally you have this much energy that needs to be put in, but now with enzymes, only this much energy. You facilitate things. Think of it. You bring this lactose, normally to break it, you would need a lot of energy to put into it, right? But all of a sudden, because you put it close together and you squeeze it in a certain way, you facilitated breaking those bonds, right? So what an enzyme does is when things bind to it, it decreases the activation energy, making it much easier and much more likely for this to happen, right? So here, for example, you see that without an enzyme, you have a big activation energy, meaning you need a lot of energy to break bonds down. But when you have an enzyme, you have a much more easy time breaking those bonds. You have a much easier time reaching the transition state, this energy of activation, since it's smaller, right? It facilitates your breaking of the bonds and it goes down. So, for example, here you have lactose that could be broken down into glucose and galactose. Here, normally in your body, it takes this much energy to do that. That would mean that if you left it on its own, it would take billions of years to break down on its own. So you would need to put in energy to push up the hill, and then it would be able to break. Here, with the enzyme lactase, right, lactose will be much more likely to break apart, right? So lactose will bind to the pocket over here, and it will only have this small hump here to go over. So it will be much easier to break this apart into glucose and galactose because your enzyme put it in an ideal place. And even the enzyme will shift shapes sometimes and strain the bond, right? Do you see here how it's straining the little bond here? That will strain the bond and make it much more likely for the bond to snap, right? And so this is why the EA is important and why enzymes decrease that activation energy. All right, last biomolecule to go through is nucleic acids. Woo. So we saw carbs, proteins, lipids, and now we're on to the last, best for the last. Nucleic acids, their general function of nucleic acids are, over here, there is DNA, which its function is to store genetic information for protein synthesis, okay? So actually, you know how the workers of the cell are proteins, correct? We need instructions to make proteins in our body because proteins are so important and a weird order of amino acids. We need instructions about how to do it. So this is like the archives that the DNA tells you what are the instructions to make this. I think of it like a cookbook. It gives you the recipe, right? And RNA is actually used for protein synthesis. We're going to see that this is like a photocopy of the recipe and that it will be used to make the actual protein. We'll see that in a bit. But any information, right, is how to think of this. It's a storage place for a bunch of info. And then you send it out, that information, to actually build the protein. Now, ATP is another nucleic acid, and we're going to see that this is for energy storage, okay? So this will store energy like a battery, right? We make all this energy, we store it in a battery, and then we can use it at night when the sun's not shining. So batteries, these are energy storages in the cell. Before going through this, right, I want to go through the monomers and polymers, right? So what are nucleic acids made of? Nucleic acids are the biomolecule polymers. These are nucleic acids made of building blocks of nucleotides. So a bunch of nucleotide monomers come together, A's, T's, C's, and G's, come together to form nucleic acids. You might remember these A, T, C's, and G's from high school. We'll take a look at those in a minute. So I want to start with the monomers called nucleotides. Then we'll look at DNA, nucleic acid. The monomers, these are called nucleotides, right? Nucleotide monomers, they're composed of three components. You have, first of all, a backbone of this molecule of nucleotide, and you have the nitrogenous base. Every single nucleotide, the four kinds, all have this same backbone. You have a pentose sugar that's in a pentagon shape. You have a phosphate over here. And you have this is what changes the nitrogenous base. So this nitrogenous base gives the nucleotide its chemical identity, whether it's an A, a T, a G, or a C, or a U. We're going to see this in a minute. So this is what changes, right? Remember the R group in proteins? There was a backbone that never changed, and then there was an R group. Well, this is the same. This is the backbone, a pentose sugar, a phosphate, and what changes, right, is this nitrogenous base over here. So, deoxy, this is the monomer. Now, deoxyribonucleic acid, DNA, and ribonucleic acid, RNA, are different in the nucleotides that they have, okay? That's why there's two types of nucleic acids. The pentose sugar in DNA, right? You have this pentose sugar that is a deoxyribose. In RNA, all of the different nucleotides have this ribose as a sugar. Okay, so that's one difference between DNA and RNA polymers. The second thing is that the nitrogenous base, this thing over here, if you have two different categories of nitrogenous base. You have pyrimidines, which have a single ring, and you have purines, which have a double ring, two rings, right? This says creepy tombs under the pyramids, right? So pyrimidines, right, has C, T, and U. Purines, pure as gold, has A's and G's. So the pyrimidines have cytosine, C, thymine, T, and uracil, U, right? So the pyrimidines are C, T, and U. Now, the thymine is only in DNA. The uracil is only in RNA. That's something that's different between DNA and RNA, okay? In purines, we have adenine and guanine. This is found in both DNA and RNA. So these are the purines that we have. So these are the building blocks. These little things here, they just pretend that you slip it and you put it in this place. There are 1, 2, 3, 4, 5 different types of nucleotides that you can make because there's 5 different nitrogenous bases that you could put in this position. Okay? So, formation and breaking of nucleic acids. You can also do a dehydration synthesis, linking of nucleotides together to form a nucleic acid polymer. You will take this nucleotide, which has a phosphate. It has this sugar, and it has this base, nucleic acid base. Notice I just put C here, but you know that it would look like this, right? So this is just, I'm writing C. This over here is another nucleic acid, phosphate, sugar, and over here you have T, right? So that's thymine over here, right? So in our DNA sequence, right, we're going to have C and then T. We want to build that DNA sequence. Sometimes we build up our DNA and we replicate it. So this is a way that, for example, that we would build it. We want to link these two through a dehydration synthesis reaction. Well, we're going to go and take out the water from this OH and an H over here. We're going to dehydrate it, and we will form this phosphodiester bond. We will form a bond between these two nucleic acids. I'll talk about what phosphodiester means in a bit. So this is forming a nucleic acid from two nucleotides. You could do the opposite. You could break a DNA down or an RNA. You could take these nucleic acids and break the bond here. This is called hydrolysis. You would need to insert a water, and this would form two separate nucleotides, right? So that's called a hydrolysis reaction because you need to insert water to break it down. Nucleotides, when they link together, we don't, remember in carbs, we called that a glycosidic bond. In proteins, we called it a peptide bond. In nucleic acids, we call that a phosphodiester bond, because the phosphate is going to be linking the two together, and on each side of this phosphate is an ester bond, diester, because there's two ester bonds. You don't need to know about ester bonds, that's chemistry, but I need you to know that between these different nucleotides are phosphodiester bonds that are formed, okay? So one other thing to know about nucleic acids is they actually have directionality. Do you see over here, we're going to look at one of these little bits here. So this yellow thing is the phosphate group, right? This little green thing is the sugar, the pentose sugar. And this little orange thing is the nitrogenous base. It could be A, T, C, G, whatever, right? In a nucleic acid, this is one nucleic acid, right, with many nucleotides that are in it, right? So this is one nucleotide monomer. This is an entire nucleic acid polymer, right? And this is what we call the sugar phosphate backbone. This is the phosphate, sugar, sugar, phosphate, sugar, phosphate, sugar, phosphate. That's the backbone that keeps repeating. What changes is the nucleotide. This could be A, T, C, G, right? It might change up. So that's our polymer over here. I want you to notice that there is, we number our carbons on our sugar, right? This is the first carbon, second, third, fourth, and fifth. The fifth carbon is always the one that touches the phosphate. The first carbon is always the one that touches the nitrogenous base. So technically, here is a phosphate. This is like, we call this end the 5' end of the DNA, because the phosphate is hanging out over here. If you go down to the very end, this is the phosphate here. That's the 5' carbon. And over here, right, you have the 3' carbon that is hanging out over here. So actually, this 3' carbon is just hanging out. We call this DNA, it has a direction. We say that there's a 5' end and there's a 3' end, okay? So polymers. I talked about the monomers, right? This was what was important about the monomers. There's a sugar, a phosphate, and a nitrogenous base. Now we need to talk about the polymers, the nucleic acids. The first one to look at is deoxyribonucleic acid, DNA. Then we'll look at RNA, then ATP. So DNA is a double-stranded and complementary nucleic acid, okay? So this is a single strand of the DNA. T, G, C, T, G, C, A, T, C, T, G. Do you see all these letters repeating? Right? That's a strand of DNA. This is one monomer, another one. We're going to often show it like this, but you know that in this strand, this G over here, right, is a phosphate, a sugar, and a G, nitrogenous base. You know that this A over here is a phosphate, a sugar, and an A, nitrogenous base. This T is a phosphate, a sugar, and a T, nitrogenous base, right? So that's the single strand of DNA, right? Cartoons often show it like this, just to simplify things. But you know that this is the monomers inside. One monomer, 2, 3, right? The truth is, this is single DNA strand. It's not like this in our body. In humans, we have 2 DNA strands that look like this, and they form a double helix, right? One is in one direction, the other is in the opposite, but they bond in the middle through weak bonds. We'll talk about this in a bit. And they actually twist. If you take these 2 strands and then twist it, they form this DNA, right? You might have seen in the movies. It looks very cool, but they actually do twist like this. The two DNA strands are not identical, right? This strand here is not the exact same as this strand here, but they are not identical. This is a T and this is an A. Not identical, but they are complementary. Nitrogenous bases pair with each other via weak hydrogen bonds, which point bases inwards and form the stabilizing double helix, right? So the bases point inwards and the sugar backbone is on the outside, so it's very stable helix. What I want you to know is that what binds to the other one is always complementary. A's will always bind with T. Always. Because this A, right, can make some hydrogen bonds, some weak bonds here with this T. And G's will always bind with C's, right, because they're complementary. So you see over here, T always is with A, G with C, C with G, T with A, T with A, C with G, G with C. You always have these pairings, always, right, in DNA. So I want you to pause the video and take a minute to do this problem. So over here, right, you have circle one nucleotide. Let's go circle one nucleotide. Be careful. I could circle one nucleotide kind of over here, right? But that's not a very good picture. Actually, I'm going to take that away because this isn't the best place to circle it. Where I want you to circle it, this is a zoom in of this section of the DNA here. One nucleotide, we know, has a phosphate, a sugar, and a nucleic acid, right? So this is one nucleotide. Oops, I should get that hydrogen there. Let me undo that. Let me go and undo that. I will go and circle this hydrogen included, right? This is one nucleotide over here. Oh boy, bad circle. But I wanted to include all the aspects. Do you see all of this here? That's my nucleic acid base, right? Sorry, my nitrogenous base is over here, right? I have my sugar and my phosphate. Remember, a nucleotide is made up of a sugar, phosphate, nitrogenous base, right? And we have those three elements here. Just for fun, right, I will do it with the other ones. So this is my other. I could have circled this as my nucleotide. I could have circled this as my nucleotide. Or I could have circled this, right? Okay. I could have circled this over here. I could have circled, oh, sorry. This is missing a phosphate. Let me undo that. I will circle this over here because I need a phosphate, a sugar, and I need my nitrogenous base. I need a phosphate, sugar, nitrogenous base. Phosphate, sugar, nitrogenous base. Phosphate, sugar, nitrogenous base. So I could have circled any of these and that would be correct for this answer, but I should just circle one, right? Alrighty, so let me just keep one, right? This is going to be my answer that I chose, this one here. Circle one, so this is my nucleotide. Circle one nucleic acid. Here is one nucleic acid, right? This here is one entire nucleic acid. It is bonding with a second nucleic acid, right? There are two separate single DNA strands that form one big DNA molecule that has two strands, right? DNA, we say, is double-stranded. So there's one nucleic acid here, right? And one nucleic acid here, right? Okay. Let's just remove that. For the sake of my answer, I'm just gonna circle one, right? I'm just gonna circle this to keep it simple. Where is the backbone of each strand, right? The backbone of each strand is the sugar phosphate, not the nitrogenous base. So this arrow here, right, this gray arrow, actually shows the sugar phosphate backbone, right? This is the backbone of each strand. On this side, it's over here, right? Do you see how it's always a sugar, a phosphate, a sugar, a phosphate, right? And these are the actual bases. They're not part of the sugar phosphate backbone. Name the bond that links nucleotides together. Nucleotides, right? This is one nucleotide. This is another nucleotide, right? Oops. They are linked together by what? They are linked together by this bond, right? That's called a phosphodiester bond, right? Phosphodiester. Oh God, sorry about my writing. Name the bond that links the nucleic acids together, right? Well, this one is one nucleic acid here and another nucleic acid here. This is the bond over here that links the two together, right? This is called a hydrogen or H bond, right? Hydrogen bond or H bond, because usually a hydrogen's involved and they're weak bonds, okay? Why is DNA double-stranded? Because it stabilizes the molecule and it hides the bases inside, right? And the nice big strong bond, the sugar phosphate on the ends protect the outside, and they protect the inner nitrogenous bases that are there, right? Okay, that was DNA, right? And I want you to notice something. G is always paired with C. T is always paired with A. C is always paired with G. A is always paired with T. Okay? Now, let's look at ribonucleic acid, RNA. All right? RNA polymers contain ribonucleotide monomers, right? Ribonucleotide, these are not deoxy ribonucleotides. These are ribonucleotides. So these over here, remember the difference between DNA and RNA. We said that it still has a phosphate, still has a sugar, but the sugar is not deoxyribose. It is just ribose. So deoxyribose occurs because this little H had an oxy, right? Had no oxygen, sorry, and this ribose does have an oxygen. So this is ribose, which has an OH here. Deoxyribose does not have an O. D means not, oxy, no oxy, right? But this is still a ribose sugar. So this ribonucleic acid, the only difference really with the sugar is that this is an OH and it's not, in DNA, it would be just the H, okay? So let's erase that. What we have right over here, so we have ribose. What else is different? So phosphate is the same. We still have a ribose, but it's just that it's ribose and not deoxyribose. The base here, right, is, remember in DNA we said it was A, G, T, or C for DNA. The nitrogenous base for an RNA, right, we have the pyrimidines, which are C and U for RNA, but in DNA it was only C and T, right? So now for RNA, we do not have T anymore, and we actually only have U. So we have C, U, A, and G. Those are the ones, the bases that could be here. So what type of strand is RNA? RNA doesn't contain thymine, but instead is replaced by uracil. So in DNA, C and G pairs together, right? Blue and yellow are the ones that are always pairing. A and T always pair together in DNA, right? Well, in RNA, C and G still pair together. You see C and G always that are going to be there, but A and U are the ones that are pairing together because U is replacing the T, right? And the second thing to notice is that RNA is single-stranded. It's just one strand of RNA, and DNA was double-stranded, okay? So what is the function of RNA? DNA is in the nucleus, in the special castle in the cell, and it has all the instructions to make our proteins. I like to think of it as your grandma's very important cookbook that she has at home, okay? This has all the instructions on how to make her amazing recipes. RNA is used as a messenger. The cell transports an mRNA, a copy of the DNA instructions, out of the nucleus into the cytoplasm to build a protein. So here's grandma's amazing recipe that she always makes for you. This is her cookbook, right? She's not going to let you take that home, right? Instead, she says, go make a photocopy over here, right? And go to your own house. So you make a photocopy of that little important section, and then you take it out of the house to your house, and then you could build the protein, build the recipe at home using the instructions, right? So DNA are instructions in general. mRNA are photocopies of the instructions so that you could take it out in the outside of the nucleus and actually build that protein that you need. So, okay, let's go and do this. The difference between DNA and RNA. I want you to pause the video and fill this out. Alrighty, so over here, right, the differences between DNA and RNA, you should have had that the type of strand in a DNA molecule is 2 nucleic acid strands versus in RNA it's only 1. The location and function, right, DNA is stored in the nucleus and it stores an organism's genetic info, whereas the RNA, right, has many more functions, including it acts as a blueprint for the proteins. It acts as the photocopy to make the proteins. The sugar in the backbone is called deoxyribose, whereas in RNA it's called ribose. And the nitrogenous base, right, they are in DNA, it's adenine, guanine, cytosine, and thymine, AGCT, right? In RNA, it is adenine, guanine, cytosine, but in this case, it's uracil for the RNA molecule. All right. Last but not least is cellular energy nucleotide. So we have a nucleotide that is a bit different than just adenine. We have adenosine triphosphate. And we have adenosine diphosphate that we're going to take a look at. So, what is this? Over here, nucleotides like ATP can act as energy sources. Adenosine, right, is adenine plus a ribose plus a phosphate. We saw this. Phosphate, sugar, and the nitrogenous base. Adenosine diphosphate, if you add another phosphate over here, right, this contains 2 phosphate groups over here. That is adenosine diphosphate. This is what we call the spent version of this molecule. It's like your wallet is empty, okay? We'll talk about this in a bit. Adenosine triphosphate, if I add another phosphate here, it contains 3 phosphate groups. 1, 2, 3, adenosine triphosphate. This contains 3 phosphate groups and it's charged. It's high energy. This is a good energy molecule for you to do work in your cell, okay? And I'll explain why this is. An arrangement of the ATP. I want to look at this ATP, very important molecule in our body. Is very similar to the nucleotide in RNA with simply 2 additional phosphates. So this is normally, minus these 2 here, this is normally adenosine, right? But if I add 2 phosphates, then I get ATP. And I want to explain to you why it's a high energy molecule. Do you see these negative charges on the phosphates? These are negative charges. When you bring 2 negatives together on a magnet, what happens? They will repulse, right? They do not want to be next to each other. So if you stick this negative close to this negative in a bond, you need a lot of energy to make that bond, right? And so they will want to push against each other. They have a lot of energy next to each other. If you put 2 magnet ends that are similar close to each other, that's magnetic energy trying to push them apart, right? So there's a lot of energy in these bonds because of the negative charges. So when you have ATP, you have 3 negative charges that are all pushing against. These are very high energy bonds. So when you have 3 phosphates, ATP, this is charged, it's high energy. You have a lot of negative charges pushing against each other. When you remove one of those bonds, that's ADP, adenosine diphosphate. You remove one and then there's less energy there. You removed one of them and you split it apart. There's no more negative pushing, so you removed a high energy bond, okay? So I want to talk about the ATP-ADP cycle because this allows energy to be made and used in our body. So the first thing is over here, right? When we produce energy in the bonds of glucose, we eat glucose, right? In our body, we ingest and absorb the glucose, and glucose is catabolized and broken down during cellular respiration in our cells. Our cells of the body break down glucose. That's why we need to eat, right? This happens in the mitochondria, and this process will release energy. So when we break down glucose, we release energy in our cells. And our cells, the mitochondria in our cells, takes this ADP, this wallet that is empty with 2 phosphates, right? ADP, adenosine, because this is adenosine. Diphosphate, it only has 2 phosphates. That's adenosine diphosphate, the spent version. It takes your empty wallet, and because you worked and you burnt glucose, right? You got energy from cellular catabolism, from breaking down the glucose. You got energy, and that will add a phosphate onto this ADP. Do you see this? ATP now has 1, 2, 3 phosphates, right? So that's ATP. So the energy released turns to spent energy. ADP gets converted into ATP, which is a molecule with chemical energy that's stored in its bonds. There are 1, 2, 3 phosphates here, right? And there's 2 very high energy bonds. These phosphates don't want to be next to each other. This is a high energy molecule. This is like you got your wallet and now all of a sudden you have money in your wallet, right? So you filled up your wallet with money. Then you take that money that you got from work, right? And you will then go use up the energy. You will go spend your money. Energy is spent from ATP by tagging its high energy phosphate onto another molecule. So do you see this ATP here? It has this phosphate that has a lot of energy. It will now go and tag it onto another molecule to pay for something, right? And this is called phosphorylating it. So here, for instance, I have my ATP with lots of energy in it, and I phosphorylate. Actually, the ATP gets dephosphorylated. It removes one of its phosphate, but it phosphorylates another protein. So this is a dephosphorylated protein, and this is the process of phosphorylating this protein, where this protein now has a little P on it, a little phosphate, right? This protein will have the phosphate, and so ATP took off its phosphate and put it onto another protein so that it is now a high energy molecule and it could go and do work. This protein can now go and transport something or break something, right? So this is a high energy molecule now. And so now your wallet, though, is empty because you used it up. So our cells of the body, right, when you eat glucose, that fuel for the body, you don't use it for work right away. You don't spend the energy from it right away. This is kind of like us in society, right? We will go to work. We each have a profession. I'm a teacher. We have some pharmacy tech students, right? I go to work and I teach, right, to students, and I get paid, right? And then I go to buy medication for myself, and I go and pay with money to get the medication, right? I don't go up to the pharmacy and say, hey, I'm gonna teach you and in return you'll give me the medication, right? That would be too complicated of a system. We have a currency. That's why I like to call ADP and ATP a currency of the cell, because it's actually it's a way to exchange things, a common way to exchange things in the cell. So same situation over here, right? When you eat glucose, that energy gets converted into a dollar bill, into ATP, which has a lot of energy, right? So then the cell can use this ATP to go and do its job, right? Whatever it needs to get or buy or do, right? It will go and send that money somewhere, and it will convert. It will put that phosphate onto the other thing and pay using ATP to do that. So it's kind of a currency of the cell, we call it. And what is ATP used for, right? This ATP is broken down into phosphate and ADP because we will use it to synthesize genetic materials, to build DNA. Remember, it costs a lot of energy to make things, to synthesize cellular components, to make things in the cell. Remember that building of things takes energy. To move muscles, we need ATP. For molecule transport, sometimes we need some ATP. For electrical energy in the neurons to send signals. So all of these things are needed. We need ATP, hence why we need to eat food, break it down. The mitochondria converts that into ATP, which will then be used to do work, right? That's why we need to eat, because we need energy to make ATP, to make dollar bills, to then use the dollar bills to do things in the cell. These are the things we do in the cell that need money, ATP. And that's all for this lecture.